Hello everybody, and welcome to this advanced community ecological data analysis webinar using vegan. I'd just like to take the opportunity to recognize that I'm broadcasting this webinar today from Treaty 4 territory in the Treaty 4 lands in Saskatchewan in Canada. These are the territories of the Cree, the Saulteaux, the Dakota, Lakota, Nakota, and the homeland of the Métis. Today, these lands continue to be the shared territory of many diverse peoples, including me. Okay, well, thank you all for coming. I am a research scientist in the Institute of Environmental Change and Society at the University of Regina in Saskatchewan in Canada. I'm also an adjunct professor in the Department of Biology here, and I am also one of the maintainers of Vegan. And we had an introductory webinar on about using vegan for unconstrained ordination on Tuesday, and today we're going to look at more complex kinds of analyses where we actually try to build models using vegan and using the different constrained ordinations, and then how do we test those models. Okay. So, yep, welcome everybody. Well, over the next couple of hours, we're going to have a bit of a deep dive into some of the model building tools in vegan and how we do permutation tests and how we set up more constrained permutation tests that can handle the kinds of problems and data types that we have in ecology. Okay, so just some logistics before we get started. The slide deck is at this URL here, bit.ly/advancedvegan, adv-vegan. And the sources for these slides, if you do want to have a look at them after the webinar, are up on GitHub. If you'd already loaded up the slide deck, then it might be worth just doing a refresh now, just reloading the pages, because I just pushed some changes up to the website a few minutes ago. If you want to follow along, then there are a couple of data sets that we're using today that don't come with vegan. So you can, there's another bit.ly link there to Advanced Vegan Zip, this other one, and if you go and click on this, it will actually download a zip file to your computer. And then if you unpack that and go to the folder where that was unpacked and open up the .r file, then hopefully RStudio or your editor will be in the right folder. And then the data sets are in there. And this is just a copy of everything that's in the Git repo. Yeah. So if we're going to try and do some question and answer, because there were so many people that were interested in these webinars, I've not been able to do them via Zoom. So what I'm doing is having this Google Doc where you can put in some questions, and I'll do my best to answer those questions as we go along. I may not have time to do answer all the questions, but I will do my best. I've added sections into the Google Doc, so put your question under the relevant sections, and when I get to the end of a section or I need to take a slight break, I will stop and go and have a look at those and see if there's questions that have come up. Okay. Just to remind you that this live stream is being recorded. It seems to take YouTube a little bit of time to render the high definition version after the live stream is finished. So and after I've cropped off the stuff from the beginning before we went live. So it might not be today that you get an email, but I will email you with the recording, and I will update the GitHub repo that's up here. I will update that with the link to the recording when it's available. But I will also email you. It just might take a day or so for for YouTube to finish doing the processing. Okay, so what are we going to talk about today? Well, we're going to start off by looking at constrained ordination. And we'll look at the two main techniques for this, canonical correspondence analysis and redundancy analysis. And then I'll explain this idea of doing partial constrained ordination, where we remove the effects of some variables, perhaps some nuisance variables, and then we will look at the effects of the remaining variables. We'll then look at model building and how you sort of build models in vegan, and this should be familiar to you if you've used any kind of the regression tools within R, but we'll go through that, and then how we do model selection, and I'll talk a little bit about the forward selection ideas that are available, and where we have to be a bit careful with doing model selection and model building in vegan. And well, with constrained ordinations in general, it's not just vegan. And then we'll look at permutation tests and what vegan is actually doing when it's doing a permutation test. And then we'll look at the restricted permutation tests that allow you to handle a more diverse array of problems than you can with the simple randomization. And then we'll do PERMANOVA. Several people have asked about this, so I've got some slides on PERMANOVA. And then if we have time, we may not, I'll look at the diagnostics, some tools, but they're in the slides. We'll see if we have time to get there. Okay. Okay, so we're going to start with constrained ordination. And we'll just start with CCA, this canonical correspondence analysis. So canonical correspondence analysis or constrained correspondence analysis is sometimes how you'll see it in the literature. It's the constrained form of correspondence analysis. So if you were familiar with correspondence analysis, you know that fits these unimodal curves following a species packing model. It's basically just a weighted form of principal components analysis, but it finds axes, correspondence analysis finds axes that best separate your sites along these axes, assuming that your species are going to be responding unimodally. Okay. What canonical correspondence analysis is doing is saying rather than just finding the directions in this multivariate space that best separate your sites, and then looking afterwards at how if you can explain those latent variables with the environmental data that you've collected, what CCA instead is doing, it's a constrained ordination technique. It will only find axes that can be explained by your environmental data. So the fancy way of saying that is to say that it finds axes that are linear combinations of the environmental data. Rather than in CA, the unconstrained form, we find linear combinations in the species, these latent variables. In CCA, the axes that we form have to be explainable by the environmental data that we collected. Okay. And when we come on to redundancy analysis, that's just the constrained form of principal components analysis. And it's the same thing, except that's really more useful for linear things that respond linearly over gradients, like environmental data. Or, as we saw on Tuesday, you can transform your data in certain ways with certain transformations, and then the RDA reflects more these non-linear patterns because you're not now recovering the Euclidean distances, which are useful for sort of linear things, but it might be some other dissimilarity like chi-square or the Hellinger distance. So there are two interfaces to fitting models, constrained model. The main two interfaces actually for fitting the models in vegan is to use the CCA function if you are, so the CCA function if you're doing canonical correspondence analysis, or we use the RDA function if you're doing redundancy analysis. They work the same way. There are two interfaces. There's the basic interface where you just give it your two data frames, and somewhat confusingly, back in the day, these ended up getting called X and Y. X is actually the responses, so it's back to front to how you might have seen in regression models with Y being the response. Here, X is the response, so you give it a data frame of your species data, for example, if that's what you're trying to explain. And then the Y matrix is your matrix of constraints, or a data frame usually. Okay. Now, that just can get confusing, and you'll see later there is also a Z matrix as well. But it's better to use the formula interface, and I'm showing this here. The only difference between the formula interface really for CCA and RDA is that we put the entire data frame of our response variables, in this case our species, we put the entire data frame over here on the left-hand side of the formula. And then if you want to do the really bad thing, which is to fit a model with all variables, all of your environmental variables, then you just put this dot on the right-hand side of the formula. And that just means give me everything from the data frame specified by the data argument. So this, you have a data frame on the left-hand side, and then the dot just specifies all of the variables that are inside var chem. more, I mean, I've just written this for space, but you'll see in a minute what we really want to be doing is actually specifying a model properly here and stating which variables should be included. So the formula interface is actually, I think, more powerful because you can include interactions much more easily that way and various other things. So it's the recommended interface to these ordination techniques. And this is what it looks like when you do that in Vegan. So if you're wanting to follow along, I think I've missed something off on my slides. There are a couple of things you need to do. One of them is to load the data and load the Vegan package. And if I just go over to my Google Slides for 1 second, I just need to move things out of the way. So the Google Docs, I will just add something in here. So we need to do library Vegan and then data var chem, comma, var, can't type, var spec. Okay. And then if you run those 2 lines, then you'll, and they're in the Google Doc now, then you'll be able to follow along with what I'm doing on the slides here. I'll be going quite quickly because a lot of this is sort of, perhaps sort of people will know, and it's very similar to the sorts of stuff that we had in the intro vegan lecture. So I'm going to be going quite quick for a minute. Okay, so this is what we get. We get, when we do that, we get an output from the model. So what model did we fit when we did this, just to remind you what that object is. We get some summary about the variance, the total variance in the data set and how it's partitioned. So we have the total variance here. And I'm using variance in a loose term. It's actually, we refer to it as inertia consistently across the ordination methods in vegan. So I'll talk about inertia actually, but then each of the outputs will tell you what the inertia is actually a measure of, okay? So in this case, in CCA, it's actually a scaled chi-square metric. But that's the sort of total amount of variation in your data set. This is the line that says the constrained. That's the amount of variation that was explained by all of these environmental variables in this data set. And this line here, the unconstrained, that's the amount of variation that's left over. And so we had 14 variables in our model, and then we have 9 remaining axes of variation. And what these are, this remaining axes of variation are essentially, if you've taken the residuals from this model and then use them as the responses and done a correspondence analysis on them, that will show any structure that's left over in the residuals. And ideally then you would want the eigenvalues for those unconstrained axes to not really be explaining much variation at all. Okay? They'll have to explain some because this mops up all the remaining variation. But you would hope that there wouldn't be any major structure in here. If there is major structure in here, it suggests that you have variation that isn't explained by your, well, it literally isn't variation that's explained by these variables in your model. And so you may have missed something, or you may not. These are all going to be linear effects of these variables. So the effect on the species, on the responses, might be in some way nonlinear. And that hasn't been captured because you fitted a whole load of linear terms here. Okay. So we get this summary. You can work out then what the proportions are. So with all 14 variables, we explain about 70% of the variation in this data set, with about 30% left over. And redundancy analysis works in the same way. We get exactly the same kinds of output, except now we have a different measure for the inertia. And because I didn't standardize any of the variables that went, any of the species variables that went into this, this is now, inertia is variance. And so we have these very large variance values. And this is one difference between Kanoko, if you've ever used that software, and vegan. Kanoko would standardize this total variance to equal 1, and the eigenvalues then would be scaled accordingly. We don't do that in vegan. We just sort of, this is the actual variance and then the proportions. And in the webinar on Tuesday, I showed you how we could go through and get the sorts of outputs that vegan would give you, that Kanoko would give you, right? But these models work the same way. Essentially, it's just the underlying response model that we're saying is different. In redundancy analysis, we're saying that the responses respond linearly along the gradients, and the gradients are linear combinations of the measured environmental variables. And in canonical correspondence analysis, in CCA, we say that the species respond unimodally, according to some species packing model, along the gradients, and the gradients that we extract have to be these linear combinations of the environmental variables. Now, I've said a linear combination, but just as in regression, you could have a linear combination of these variables and linear ones. We could put interaction terms into this model. We could put polynomial terms into this model. We could put splines into this model. In the same way that you would have done with and factor variables can go in here as well, that would go in just as like you would with an ordinary regression model. So it all works the same way. The only thing, when I said splines, for those of you who know that I spend a lot of my time fitting generalized additive models, you can't use splines from MGCV in here. You'd have to use sort of like the natural spline or the B-spline from the splines package in the formulas. Okay. So I'm just going to go through a few things. Often when people are very familiar with these methods, they want to go and start taking bits of information from and getting bits of information that they might be familiar with from other software and then doing things with them. So I want to spend a few minutes just talking about the CCA object, and for various reasons the CCA object is called the object that you get back from RDA and from CCA and from cap scale and from distance-based redundancy analysis in vegan all inherit from this class CCA. It's just for various reasons. CCA was the first thing that Jari, one of the first things that Jari implemented in vegan, and he just thought he'd only implement CCA, and so he didn't want to change the structure of the objects when we added in RDA and other things. So it's just an artifact of history. So all these objects inherit from this class. Essentially, you can look at everything that's in it by going to this help file. Depending on what kind of analysis that you do, some of those will be null. So like if you've done a partial ordination, then the partial section will be filled in, but if you haven't, then it will be null. Okay, so they're there, it's just some of these things will be null. And we're just going to go and have a look at some of these things that are available to you. So loads of things. We've got the call, grand total, that's just the sums of the row sums, so it's like the total sort of chi-square value. Or rather, the total weights, I guess. We have the row sums and the column sums, and these are the weightings that are getting applied, for example, in the CCA. We have the total inertia. We have these three components then that contain the different scores. We have the partial, the PCCA bit. Those are the bits that were partialed out. So that will be the information related to the how much variation is explained by the things that we removed from the model before doing it. Then these are the scores associated with the regression part of the model, and then these would be the scores that are associated with those unconstrained components, the residuals that are left over. Okay, and then there are some other bits in here in these objects, and if you know what you're doing, you could go and extract these things and work with them, but in the same way that you would extract bits and pieces out of a linear model. We'll say you have to be very careful about these things. There is no guarantee that we will keep these things named as for CCA because it's confusing. If you fitted a redundancy analysis or something like that, PCA, that they would have these components that are named for the unimodal method. So those might change. And so one thing that you might want to be a bit careful about is just going and extracting these scores directly from here. Okay. So basically I've said most of this stuff here. Eigenvalues and the axis scores, because they're spread out between these different components, and we have an extractor function that will allow us to extract this information much more easily if you want to get out the scores or the eigenvalues. Okay, so if you want to get all those eigenvalues out, so only some of them were printed before, then the eigenvals function can be used to extract those eigenvalues, right? And there's an argument in here that you can control what you extract, whether you're extracting them from the constrained part or the unconstrained part, for example. So I'm going to work through a quick example now. I'm going to fit a CCA model to the lichen pasture data that we've just been looking at. And the model is going to include the nitrogen, phosphorus, and potassium. Those are the only things that we're going to put in the model. I'm going to save that under a particular thing, and then I'm going to look at how much variation is explained by this model, and then I'm going to extract the eigenvalues from this model as well, and look at how many constrained axes there are, just so that we get a feel for how we do this analysis. So the first step, and this is what I'd got in here. I should have had it earlier. But the first thing is to load the package, load vegan using library vegan. If you haven't got vegan installed, then you'll need to install that first. And then we use the data command here in this instance to load the data from the package, because at the moment we're going to use these data sets from the package. Okay, so this is then how we fit our model. Notice now that I'm being explicit about the variables that I want on my, I'm being very explicit about the variables that I want on my in my model here. Okay, so we'll look for those 3 variables in this data frame. I stored it in my object, and now I just type the name of the object to print that out, and here we get a summary of what's been fitted. And so we can see that there are about two and a bit amounts of variance in here, and we can explain about 21% of that. So we explain about 0.45 bits of that variance, so about 21% with those 3 with those 3 constrained axes. So there are 3 constrained axes, and we explain 0.45, or in other words 21%. And so there's a lot left over. There are 20 unconstrained axes. These would be the residuals, and they explain about this sort of 78-79% of the variance. Yeah. And if we look at our constrained axes, the size of these 2 eigenvalues, they're sort of relatively similar in size, and then we have a bit of a drop off in the third one. So it suggests that we can represent most of this variation. We can represent like 0.26 of this, 0.4. We can represent about 0.26, so the sum of those first 2 eigenvalues. We can represent a large part of that with just the first 2. We perhaps don't need to worry so much about this. This is going to be explaining just small amounts of the variation remaining. But then also notice that the eigenvalues for the unconstrained parts are much larger. Certainly the first 2 are much larger than any of the eigenvalues that we got for our constraints. And what this is telling us is that there is still a lot of structure left in our residuals. So if you got something like this, this would be telling you that either there are things that you haven't measured, that are, if N, P, and K were the only things you'd measured, then this would be telling, okay, there are lots of other things going on in this data set that you haven't measured, or that the effects of N, P, or K are not just additive and linear, as we've assumed here. So we might want to put in interactions, you might want to put in some non-linear things as well. And that will be should be driven by theory. Okay. So we can extract those eigenvalues, and here I'm showing you how to extract the eigenvalues, just the constrained eigenvalues, using the model argument. So we just pull them out and we look at those. There are just 3 of them, and we can look at the length. There are only 3 of those. So I didn't actually need the head thing here. I was just getting, I'm just so used to putting that in because it keeps flooding my screen, my slides. Anyway, so that would have answered our questions. If that was the kind of model that you wanted to fit, we would have our 3 variables in our model. We'd fit that. We get some idea of the how much variation those 3 variables can explain, and we can look at the residual structure as well. Okay. Now I've already covered this in the webinar on Tuesday, and so I'm going to go through this relatively quickly. If this means nothing to you, then you should be perhaps go back and watch the intro webinar. So there are lots of scores that are possible, and there are actually a lot more types of scores that are possible with a constrained ordination, because there are lots of different ways that things that we can extract from these models and present them. I mean, we have 3 sets of scores, but some of those scores can be represented in different ways as well. Okay, so we have this function called scores, That's what you should use to extract any of the axis scores. If you want to do your own plotting, if you want to take this information and do other things with it, then the scores function will give you back the positions on the axes of the species, the responses, and the sites and your constraints, depending on what you ask. But you need to use this, you should use the scores function to ask for them. Okay, so you give it the ordination model that you fitted. Then you tell it which axes to return. So the choices argument tells it which axes you want, and it defaults to just extracting the first two. Right? And you can extract whichever axes you want. Okay. Then the display argument will control which type of scores get returned. Now you can just give it one, so you could display equals sites, for example, and you would get the site scores. And those would be for the rows, the observations, the samples, or the sites, whatever you want to call them. Or if you did display equals species, you'd get the scores for the species, the responses, the variables, whichever way you want to think of that. Okay. But you can also pass in multiple as well. So you could put in display equals concatenate sites, species, and what have you, and you get more back. Right. So sites and species give you back the different, give you back the scores for the observations and the variables. Then there are these things called linear combination site scores. These are site scores that are sort of closer to the regression, and these are sort of like the fitted scores given the regression model. So this is closer to the regression model, whereas these site scores that you can also extract with WA, those are halfway towards the more unconstrained bit. So they reflect the variation between sites in terms of the species composition as well, whereas these ones reflect purely the fitted values of the environment. And then there are some other scores. We can get the biplot scores, so these are the coordinates for the tips of the arrows for your explanatory variables. Or if you've got factor variables in your constraints, then the CN or centroid scores will extract the factor centroids for you. Okay. And by default, when you plot, and a lot of the tools in vegan will make some assumptions about what you want and give you back sort of what it thinks is appropriate. Okay, but you're in full control. And a lot of the functions, you don't necessarily have to use scores, but like if you use plot, you can set the axes through the choices argument, and you can also set what scores you want to display on the diagram using the display argument. So you can, these arguments get passed on to scores. So you'll find them as arguments to lots of the vegan plotting functions and other functions. So here what I'm doing, I'm just showing that I'm extracting the scores from that CCA model. I want the first 4 scores, axes, and I'm extracting here the species and the site scores. Okay, and the str command is just showing the structure of this stuff. Okay, so here I'm extracting 4 of them, so I get back a list with a species that is a component which is species for the species scores. That itself is a matrix with 44 rows and 4 columns because we wanted 4 scores, and there are 44 species. And then we have a sites component, which is there are 24 sites in this data set, and we ask for 4 axes. And these things, if you just ask for one of them, here I'm just showing asking for axes 1 and 2 and the site scores, you just get the matrix returned. You don't get this list. And so if you come from the tidyverse world, this would be frowned upon. What you get back from the scores function depends on the arguments that you put in, i.e. the type of object, either a list or a matrix. But this was all set in stone well before these ideas of trying to be consistent. So you just have to be aware of that, especially if you're programming or doing anything with it. Just remember that if you ask for more types of scores, you get a list. And if you ask for one type of score, you get a matrix. Okay, and then scalings. We've got lots of bits of information that we might want to show on these ordinations. You might be showing 2 or even 3 things if you're trying to show the species, the sites, and the environmental variables all in one go. And we can't possibly show all of those things without distorting the relationships among them and between them for some of them. And so what we often will do is we'll decide what we want to focus on in the plot, and we will scale the diagram accordingly. And usually it's a choice between whether you want to focus on the sites or you want to focus on the species. Do you want to focus on the observations and how similar the observations are in terms of their species composition, or whether you're more interested in the relationships between the response variables, the species, and the correlations and between them. Okay, and the way we specify that in vegan is using the scaling argument. And something's gone slightly wrong on my slide here, so I apologize for that. I'll have to fix that later. There are different scalings with different numbers. If you're familiar from from Kanoko and what have you, then these numbers might actually mean something to you. We have focusing on sites, species, or symmetric scaling. We have a set of negative numbers, which would give you the hills version of those. For a CCA or for RDA, you actually get correlation-based scores. And yeah, if you don't want any scaling, you put scaling equals 0. Okay. Now, thankfully, we don't actually have to remember all of that. I couldn't remember all of that and kept getting confused behind them. So a few years ago, I actually went in and modified the way the scaling argument actually works. So now you can use those numbers if you want, but I can't remember them. So I wrote this thing where you could just pass in a character vector saying what kinds of what scaling did you want. So you could have no scaling, you could have focusing on sites, focusing on species, or have that symmetric scaling, which actually scales both and tries to be this optimal kind of scaling. Or you could, and then there are two modifiers. So there's this argument called hill equals true, and there's an argument correlation, which is set to true. That will then get you those negative scores. So if you've done CCA, you can get hill scaling by passing in hill equals true when you extract the scores. Or if you've done an RDA, then you can pass in correlation equals true, and you get these correlation-based scores. And what they are is if you've just fitted your model and you didn't scale the responses, you didn't do scale equals true, then the species that are most abundant and have got the bigger values will tend to dominate because they have the largest variances and covariances. And normally what you would do there is you would use scale equals true to turn them into correlations rather than covariances. But if you don't do that, you can still get some sort of standardized scores out by using this correlation equals true. So we'll just go and look at extracting those site scores from the CCA model that we just fitted a minute ago. And we'll look at getting axes 2 and 3. We're going to do hill scaling and we're going to focus on the sites. Okay. And this is what we get if we want to do that. So we use our scores function. I give it my ordination object. I only want the site scores, so I set display equals sites here. I said I wanted to get axes 2 and 3 for some reason in this case, so I'm going to extract axes 2 and 3. And then we have to set the scaling. So because I was extracting the site scores, I'm probably most interested in the sites. So I should get the heights, set the scaling to be sites as well. And then I wanted hill scaling, so I turn on hill equals true. Okay. And then if this was an RDA, instead of hill equals true here, you would do correlation equals true if you wanted the correlation-based scores. And because we only ask for one set of scores, what we get back is this matrix of scores. Okay. Which it's actually stored as a matrix, and so it's not a data frame. You'd have to convert that to a data frame if you were then and if you wanted to do anything else that required data frames later. Okay. So the last sort of thing in this topic is to look at what partial constrained ordinations are. And partial constrained ordinations are where we remove the effect of one or a group of variables first. So we remove that information from the model. We essentially residualize the data with respect to these variables. And then we fit a model to what's left over. And so we have then some amount of variation that can be explained by some other set of variables. And then we would have any unconstrained variation that's left over them. So one of these things, this is a way to sort of remove nuisance variables. You know, for example, let's say you've had multiple people out collecting the data. You could maybe partial out a variable, a factor that said who the data collector was. Or maybe you've got multiple years of observations and you're not interested in the time variation, or perhaps you want to do an analysis that doesn't matter about the time information, which year you actually did the experiment in, for example. So you could partial out time and then just look at what's left over, all right? And if you're using the default interface, then you would use argument Z, where you would pass in the data frame of things that you would want to partial out first. And so what I'm showing here would be where we were fitting a model to the lichen species data here, which we pass in as X. My predictor variables, here I'm just showing the one variable just for space. But here this would be the chemistry saying how much of the variation in the species can I explain using calcium after I've removed the effect of pH. So I pass in some data frame here to Z that contained that pH variable. And so we would remove the effect of pH and then look at the effect of calcium on the species after accounting for the pH effect. And if we're using the formula interface, and this is the one I would really recommend, then we use the condition. There's a function that you can put into the formula called condition, and with a capital C, and in there you put the name of any variables that you want. So you could have condition pH plus potassium plus something else. Okay. So if you want more constraints, you can put them in here, just add them with pluses as you would normally do. And obviously you can have multiple variables here for the actual constraints. Yeah. But pH is the thing that would be removed. Then we'd fit a model for calcium, right? And so these two models are equivalent, except for the details of the formulas. And so if we just look at that, then we get a little bit extra information here. And so we can partition the total variation. So this number we've seen before, there's about 2 bits of variance in this data set. The conditional line tells us how much variation could be explained by pH. So that's the amount of variation that we partial out. So it's about 7%. The constraint line again tells us how much variation can be explained by our constraints, and we only have one of them here, so that's how much variation can be explained by calcium after accounting for pH. And that's about 9%. And then this is the stuff that's left over. Yeah. Okay. And then we have one constrained eigenvalue or one constrained axis, and that's because our model actually only has one constraint in it. Yeah. We don't count the partial bits, yeah, as in terms of the eigenvalues, because essentially we've removed that first, that of the effect of pH, residualized the data with respect to that, and then just gone and fitted a CCA to the remainder. Okay, so one final slide then is just to show this is the default plot. So if you fitted a CCA or an RDA, then what you get when you plot the model using the plot method is a triplot that's trying to show this 3 bits of information. I think the only nice thing that we can say about this is that it generates a plot quickly for you and you don't have to do anything other than plot the model. By default it's focusing on the species. That's scaling equals species here. We're trying to cram 3 bits of information onto this diagram, and that's just not really going to work easily. Okay, and so there's a lot of overplotting and things, but it is designed as a quick plot. And some of the tools that I talked about in the Intro Vegan, the last half of the Intro Vegan webinar, can be used to build the plots up. I'm not going to go into that here. I'm actually going to go into more of the technical details of fitting models and doing permutation tests. So go and have a look at the Intro Vegan webinar if you want to sort of spruce up these plots. But these things really are designed just to get a quick overview of what your ordination model is saying about your data. Okay, so that was model building. Let me just see if we've got any questions. Okay, I think I'll talk about CAP and distance-based RDA later. In fact, I will mention them later. We'll talk about those. We'll talk about how to select variables in a little while. I'm going to do model building in a minute. How do we deal with pseudo-replication? So that's probably better dealt with using restricted permutation tests, and we'll talk about those later. I don't like this idea, this term pseudo-replication, not least because the person who came up with it isn't a very nice person in ecology, but more it's related to the fact that there is variation in here, and it's not really replication. It's just the fact that you've got lots of data, and you need to take account of the fact that the data are clustered in certain ways. And we can do that in a similar way with the sort of random factors and things like that by including things in the model. And we'll talk a little bit about that in a moment. And then someone's asking, what is the sort of safe proportion of variance that should be explained? Well, there isn't any answer to that. It depends. There isn't a safe number. I mean, it just depends on how many response variables you have, the noise in them, and lots of things like that. Okay. So I mean, typically when I'm working with diatom data, silicious algae, where we might have a couple of hundred taxa in our data sets, then we might only get a 5% or 10% variance explained with a whole host of variables. But that's fine. You know, there's going to be a lot of unexplained variance because there's just lots of random variation going on within some of those species in the taxa. The thing to look at would be to see whether there was the effect was significant or not. And there's going to be, I think it's just got to recognize that there's going to be lots of controls on those many species, and you might not be able to explain them with your environmental data. Obviously, ideally, the higher the proportion, the better, because it means that then there's strong structure in there in the data. Okay. So the next topic is going to be model building. Okay. So the thing I did at the beginning, fitting CCA1 or RDA1 at the beginning, where I just used the dot for my formula, that's the surest way to shoot yourself in the foot with any kind of regression model. And it's important to remember that CCA and RDA are really just regression models, and even distance-based redundancy analysis and constrained analysis of principal coordinates, cap scale, they're really just regression methods at their basis. I mean, with distance-based redundancy analysis and cap scale, we're turning everything into dissimilarities first, but they're still there's a regression model underlying them. And so you shouldn't just go and throw all the variables into a regression model, and in the same way, you shouldn't just go and throw all the variables into a constrained ordination. And there are several reasons for that, partly because it's just, not thinking allows the computer to take control, and that's probably not a good idea. You've got to think about whether your variables are correlated and what have you. But there's another problem in these constrained ordinations, and that's you can only have as many constraints as you have sites or species, whichever is the smaller, right? And we have another problem, though, that as you add more and more constraints into the model, you get this ironic thing where actually the model becomes less and less constrained, and it becomes more and more like CA or principal components analysis, PCA. Because the more things that you have in the model, the closer the more freedom we have to choose the gradients, and the closer they'll come to being the same sorts of things as you would get if you had fitted the unconstrained model, right? So don't do that. A much better approach is to really think about the kinds of models that you want to fit, and think about what theory might tell you or what your hypotheses are, and include those terms in the model. The formula interface is good for this because we can think about, you might know from some theory what might expect, or you might have a hypothesis that you're testing, and that will suggest to you a particular form for the model. You can encode that in the formula, including just the terms that you want, and then fit that model, rather than throwing everything in one go. Okay? But the main take home, really, when you're model building with these techniques, is to just think about what it is that you're doing. Don't let the computer take charge. Don't throw everything into these models. You're going to get into serious trouble if you do that. Okay. And so just to show the kinds of models that you can fit, I mean, if you have any sort of background in fitting sort of regression models in R, then this formula here is saying I want a main effect of aluminium plus the main effect of potassium, and I want that to interact then with the effect, so I want interactions between potassium and phosphorus and potassium, and phosphorus and bare soil. Okay, so we have a main effect from aluminium, a main effect of phosphorus, and then a main effect of potassium, a main effect of bare soil, and then interactions between phosphorus and potassium and phosphorus and bare soil. All right. So that works out to be quite a complex model, but it's very easy to write it as in this symbolic way using R's formula notation. Okay? And when you do that, we get things. We have these 6 terms. Okay, so we've got 4 main effects plus 2 interactions, according to that one. Okay. So yeah, just think about what you want to do. Now, I don't know if this makes any sense for these data, but maybe this is the kind of hypotheses that you're wanting to test. You can encode them, those hypotheses in this formula, and then that's an explicit test, rather than just throwing everything in. Okay, so in terms of building models, we really don't have much choice than to either fit some well-chosen model or a set of candidate models that you that reflect your different hypotheses, and then compare those models in some way. Or we fit a full model of a set of well-chosen variables and then try and do stepwise selection on it. Okay. Now, stepwise selection is bad, and we should be very, very careful about using it. And in fact, we would tell you not to do that in regression. We probably shouldn't do that. But it is an option here. It's the only real option that we've got to try and do sort of model selection. And in a little while, I'm going to explain ways that we can do this in a sort of slightly more safe way, that we don't shoot ourselves in the foot. But still, just treat these automatic tools with a lot of caution. Right? You'd be better off, if you can, fitting a well-chosen set of candidate models or model that has in variables for your hypotheses and so on, and just fit that and just work with that model. Okay. So there is, if you want to do this, there is a standard function in R called step for doing stepwise selection, and that can be used because Vegan provides 2 things. It provides a deviance function and an AIC extractor function that can be used by step. AIC is a sort of a bit of a kludge for ordination techniques, so don't take this too seriously. And I'll show you in a minute there is actually a better way of doing the stepwise selection than using the step function. If you want to do things manually, then those of you who have done a lot of sort of regression-like modeling in using R, then you might be familiar with the add 1 and drop 1 commands. So the most useful one is you've got some model that you fitted, and then you might use drop 1, and it would tell you how your model would change if you dropped out 1 variable, and it would show you how, if you dropped out each variable, just one at a time though, which one got dropped out. Okay, so those things are there. And if you're comfortable with doing this model building and checking and things in R with regression, then these tools work for ordination techniques as well. Okay. Now, something else we have to be a bit careful with is the correlations between variables. If you put in lots of highly correlated variables, then you can, yeah, some of my stuff hasn't been rendered. Anyway, never mind. Hopefully that won't cause a problem. If it does, I'll just switch to my local slides when I do this in a minute. Okay, so the variance inflation factor is sort of a measure of how much the variance of our regression coefficients gets inflated by the presence of other covariates. And if you've got, when you compute the variance inflation factor, then there's lots of rules of thumb as to what constitutes high variance inflation factors. Here I'm just suggesting a couple of numbers. Other people will have other numbers, but you know, if you've got variance inflation factors greater than 10 or greater than 20, then it suggests that you've got some problems. And if they're greater than 20, it suggests that what a variable is entirely or almost entirely explainable by some other variables in the model. So if we go and have a look at this, for example, we can use the VIF.CCA function and to compute the variance inflation factors for each term in a model. So here, this was the first model we fitted that included everything. And we can see here that aluminium has got a very high variance inflation factor, which suggests that it can be explained by lots of other variables in here. And the aluminium in the soil is probably going to be related to calcium and the phosphorus and the depth of the humus layer. Because the aluminium becomes labile and available when the soils get acidified. So, you know, we can understand some of those things. But, you know, here we've got this is sulfur in the soil, I think, yeah. So we have to be, you know, that one's also quite high. So there could be some things here. Now, if you weren't interested in aluminium, you're interested in something else, maybe you would leave aluminium out of your model because it's just really explaining things that can be explained by other variables like calcium and pH or the depth of the humus layer here. So you need to be a bit careful about what you put in. And this is another reason why just throwing everything in can be problems, because this can cause all sorts of problems in the models, in that certain things will start to get large effects and look important, because they're trying to just offset the effects of these correlated variables. And it's the same problem that we have in linear regression. Okay. So in stepwise selection, I'm just going to refresh my slides and just see if that causes this. Okay, so what I'm going to do then is I'm just going to close this window and I'll just get the slides locally. For some reason the MathJax is not rendering. Just give me a second. Okay. Okay, now I'll just have to skip through where it would be useful to have had the Jaringen extra package. I could have skipped ahead. Where are we? Okay, so we just done variance inflation factors. Okay, so things are being rendered now nicely here. I'm not quite sure why it's not rendering from the website. I'll have to have a look at that. Okay. Right, so I said one thing, I mean, if you just want to make your life easy, then one thing you could do is to just fit the model that contained that sort of represented your hypotheses and just be done with it. Yeah, they wouldn't have to do any selection. But if you do have to do some forward selection, I said you could use this step function, but that uses AIC, which doesn't really exist for an RDA or a CCA model. And there was a paper, and I forget the reference now, but there was this paper that said that they could calculate something that looked like AIC, but it's not really AIC, and because there's no likelihood in these models. So a better thing is to use ordistep. And with ordistep, we have to do a couple of things. We need to define an upper and a lower model scope. So it might be, say, the upper scope might be the full model that you fitted, containing all of the variables, all the terms that you want in your model. And then the lower scope might be a null model that just contains an intercept. And then what we do is we do forward selection perhaps from that lower scope, from the null model, up to the upper scope, up to the sort of full model that we might do. And so again here, we're still not really thinking about, anything. We're just saying the full model is just all of the variables in here. But I'm starting off from some simple model, just an intercept, which actually doesn't mean anything here. And then we're going to do stepwise selection, okay? And because these involve sort of permutation tests, I'm setting the seed so that these things are reproducible. And we might also want to do I'm here for my slides, I'm setting off the trace so it doesn't print anything to the screen, okay? So that would be one way. This would do forward selection. So it would go from the lower scope, starting with nothing in the model, and it would then add the variable that best explained variance in the response, and then it would fit models to look at which is the next best variable, and so on, and keep going until we made no improvement in the model. And when you do that, with already step or with step, what you get back is a standard CCA object that has an extra component. So what it would return is this model, this object, and we called it mods, and then this is a CCA model, and it just shows the 3 best variables. In this case, these were the variables that were selected. So this model contains 3 terms. So after it added the 3rd term, then the algorithm would have stopped because we could make no more significant improvements to our model. So this tells us that with this forward selected model, we can explain about 31% of the variance in this data set. Right? And that's similar to the sort of amount of variance that we were explaining with all 14 of the variables. Right? So these variables seem to have, or be the most important ones, potentially. Now remember, aluminium had a very high VIF. So maybe if we deleted aluminium, maybe some other variables would come out, and well, they would have to, yeah, I would say. Okay, but the rest of this is the same. The only thing that we get extra is an ANOVA component, and if you look at the ANOVA component, then this tells you the ordering in which these things were added to the plot. So the first one to be added to the ordination. So the first variable that was selected out of all variables, aluminium, had the largest effect on the species, largest explanatory power. Okay. Then, given that we've already included aluminium, which is the next variable, the next best variable? Well, that was phosphorus. We add phosphorus to the model, and then we run the procedure again. We look at which is the next best variable that can explain any of the remaining residual variance in the species data. That was then potassium. And then we stopped, and you can see that potassium was right on the border of being selected. Okay? I think the default for selection is 0.05, and so that's where it stopped. After this point, once we've added in potassium, we couldn't add any more of the variables from this model that would make a significant improvement or have a significant effect when we were testing. Now, and you'll know this if you've done any stepwise selection, but it can be pretty fragile. So if we start from the full model, we don't necessarily end. So if we start, rather than I started at the from the low end. So I started at the low end of this thing with having no variables in the model, and we did forward selection. Well, we could start the other way. If you can fit the full model so if you can fit it, if you have enough sites or species to fit all the variables, then you can do backward elimination or a combination. And so this is what I'm doing here. We start from our upper scope, so we start from that full model. The scope here, I'm telling it, is that we can go from this is a lower scope, so this is a model that contains no variables, and the upper scope that we are allowing is the full model. This is just, you have to be a little bit more fancy if you're going to do this forward and backward elimination. But we're starting from the full scope here, and we're going to do the same analysis. And if we look at that, then we get this. And so now we get a model that doesn't contain aluminium. In fact, that was the first thing to be got rid of. And probably because if we've got some of these other variables in the model, then we don't really need aluminium because it's correlated with a lot of them. Okay, so now what do you do? Okay, you've got a model where phosphorus and potassium, we've got two models, sorry, where phosphorus and potassium were important, but you've got one model telling you that aluminium's important, and you've got another model here that's telling you that these other suite of variables are also important, right? And so this is part of the problem here with using these forward selection tools. There's no guarantee, there's nothing, they're not supposed to, there's no guarantee that they will actually end up at the same place. Okay, so something else that we should be doing really is to realize that if we add coefficients to our models, if we add explanatory variables to our models, then when we do that, the R squared, or the sort of measure of fit that we get, is biased. We know this from regression. If you add in a variable, then R squared will increase, even if that variable has no explanatory power at all, right? And so if you have a large number of constraints in your ordination model, then we'll get potentially a much larger R squared just due to random correlations between all of the constraints that you've put in. So if you just use ordinary R squared, it can give you a very flattering idea about how well your model fits the data. And so what we normally do in regression models would be to use an adjusted R squared, where we adjust for the complexity of the model, how many terms we have in there. And so we penalize models for complexity. And we can do the same thing for ordination models as well. And so the adjusted R squared statistic that's being suggested is this here, where we're R squared's in here, right? But we're applying a correction that is based on n-1, which is the number of samples minus 1, and then the number of samples minus the number of constraints. So this is a measure of the relative complexity of your model compared to the number of observations, right? So if you have a very complex model for the number of observations that you have, then you pay a stronger penalty, all right? And this will give you back an R squared. And this works roughly up to about a value of m, so it probably should have been a lowercase m, where the number of constraints is about half your number of observations. And if you are putting in more than m constraints, if you're putting in more constraints than half the number of observations, then this adjustment for the complexity becomes a little bit, it becomes sort of conservative and doesn't really do a good job. Okay? It also can be negative, and that just basically means that the set of explanatory variables you've got is either none of them have any real effect, or you've just made a really complex model, and that model is very complex and you probably only need a few of those terms. Okay, and so just in the same way that you know, you can have an adjusted R squared in regression can be negative, you can have the same thing happen here. And so you can compute this for your model using this R squared adjust function in vegan, and when you do that, you give it your model, your ordination, and you get back two values. So that first model that I fitted that included all of the terms in the model was the CCA1, and I just threw everything in there, and it had an R squared of about 0.7. It looks like it does a pretty good job of fitting the data. But actually, if you then adjust for the complexity of that model, then really the adjusted R squared drops a lot. And that's because we had a lot of predictors that weren't really needed. They weren't really explaining very much, and they were sort of redundant variables. And we've already sort of seen that from the results from the stepwise selection, because one of those stepwise selection just chose 3 variables. The other one, when we did the backward elimination, that just chose 8 out of 14 variables. And they were still doing a pretty good job of explaining the variance. So this is an inflated measure, made our model look really good, when in fact it isn't a very good model, and the adjusted R squared tells you that. So there are loads of problems with stepwise selection. It affects all of the models that you want to apply. It affects regression problems. And really, ideally, we would avoid doing stepwise selection at all. And in fact, you'd probably try to just do fit a single model that contained the terms that you needed for your hypotheses and just interpret that one model. Okay. Now maybe you want to test some hypotheses in some other way using this forward selection, and so you have to be very careful. But one way that you can make this a little bit more robust is to use a two-step or the stopping rules that Guillaume Blanchet and colleagues proposed and wrote up in 2008. And they have this two-step solution for models where adjusted R squared sort of makes sense. And in vegan, I think we've got this for RDA and for CCA at the moment. Okay, so what Guillaume and colleagues suggested was that you do 2 things. The first thing is you do a global test of all of the constraints, and we'll come on to how you do that global test in a minute. But the idea is you do a global test containing all of the variables that you think should be in your model, that full model with all of the terms that you think might be important, given your theory, given your hypothesis. And so you test that model and you see, does that model are there significant effects in there? Yeah, is that model significantly different from a null model? And we proceed if that test is only if that test is significant. So you don't start doing stepwise selection as the way we just did it. You don't start doing it regardless. you do this first test to decide whether you should proceed, and then, and only if you get a significant result in this first test, do you then do forward selection. But now we have two stopping rules for forward selection. We have the usual significance threshold, 0.05 for example, but then we also use the global adjusted R-squared. And so what we're going to do is we're going to stop if adding a variable will not mean that we add a well, when we want to add a variable, if it's not going to make a significant difference to the model, we wouldn't add it, we would stop. Okay. But the other thing that we can do is every time we're trying to add a variable, we can look to see whether the adjusted R-squared of that model exceeds the adjusted R-squared of this full model that we fitted originally of the global model. Okay. And if it does, what that's saying is that given the smaller subset of variables, you actually have better fit than the global model, and so then you should stop, right? Because all the other variables that might be added are only going to reduce that R-squared, right? So this is a way to stop you choosing too many things. And I think this just recognizes that the global adjusted R-squared isn't a perfect, it doesn't solve everything. It doesn't solve all the biases of having these extra constraints. So this two-step approach where you do the global test and then only proceed if that global test was significant, and then doing the forward selection with these two stopping rules is available in our ORDI R2 step function. And I'll just show you that here. So we have our ORDI R2 step. We're going to step from that model with just the intercept to a model with all of the variables in. I've turned off the trace, and then this is what we end up with. And so using this global stopping thing and the forward selection, we end up back with the same model that we did with our ordinary forward selection. Okay. And you can change the scope if you want as well, right? And things. But this just does all of those things that I just said to doing this. Okay. All right. So that was model building. I'm going to have a minute or two on some questions before I have to move on. Oh, I didn't think you could get an adjusted R-squared greater than 1, but I'll have to just check on that. Okay. Does it matter about which variables are inside or outside the brackets that I showed? Yeah, they do, because what it's saying is that you want the interactions between potassium and the variables that are measured inside the brackets, right? So inside the parentheses. So it's just a very specific model. So in terms of the two examples, would that make any difference? Yeah, it would, because you'd be getting an interaction in that case between potassium and phosphorus, and potassium and bare soil as an interaction, whereas in the one I showed, you'd be getting phosphorus and potassium interacting and phosphorus and bare soil interacting. Okay, so yeah, those models would actually have different interactions. I'm not actually sure what draftsman plots are. Never used primer, so not sure. How do you correct or overcome high VIFs? Well, the solution to that is to just leave out a variable. So, for example, aluminium might be, aluminium in that instance had a very high VIF. Maybe you leave that variable out. Now, if aluminium is something that you're really interested in, then that would be the wrong thing to do. You wouldn't leave out aluminium. You want to leave out something else, and so then you could just look at the correlations among your explanatory variables and see which variables are correlated with aluminium, and maybe leave those variables out, especially if those are things that you're not interested in. If aluminium was the thing that you were. Why haven't we discontinued the Stepwise selection? Well, because people want to do it, and I think if you're careful with it, they can slightly help. But yeah, people could shoot themselves in the foot, and people are going to shoot themselves in the foot. I don't think it's necessarily our responsibility to solve those problems for people. I think there can be good uses of forward selection, and they're limited, and you have to be very careful with it. For example, forward selection is really useful for prediction. Gets you models that have sort of relatively low prediction errors. So we might want to do that. So I think if people know what they're doing and are doing it properly, then they can, we should let them use the tools. And we don't, you know, in like other settings where we might do like lasso selection and things like that, we can't really do that for an ordination method at the moment. So there isn't an alternative. So that's why we haven't discontinued them. Okay. Right, permutation tests. Okay. So, I mean, RDA, if you look at the theory behind it, RDA has quite a lot of theory. It's just a principal components analysis of a lot of the fitted values of a lot of regressions. CCA is a little bit woolly. It's an approximation to sort of canonical Gaussian ordination or something like that. But even if we had loads of theory, it would probably, our environmental data, our ecological data would probably violate all of those assumptions anyway, because the theory would probably have to assume something about normality and things like that. And we, all our environmental data, all our ecological data would invariably scupper that problem, okay? So instead of relying on theory like t-distributions and then looking up from that reference distribution or from a t-distribution or a Gaussian distribution, instead of looking up what the probability of getting an effect would be under the null hypothesis, we use permutation tests to look at how much whether the amount of variation we explained or some test statistic that we derive from the model. We look to see whether the observed value of that test statistic is large or unusual relative to some null distribution that we derive by permutations. So the key thing though is when we're doing permutation tests is to decide what is being shuffled and how we're actually doing that shuffling or that permutation. That's the key thing to decide whether the test is valid. And there are several things that we can do before we actually work about how we shuffle them. We want to look at what we're going to shuffle. And so in vegan, if we have no conditioning, no partial variables, then we can just shuffle the rows of the species data because it doesn't really make any difference, right? And so that would be the, I think it might be model. I think the argument might actually be method. I've forgotten now. That would be the direct method, okay? If you do have partial or conditioning variables, then you can't just shuffle the species data, because if you do that, you break the relationships between the species and the partial variables, or the relationship, as well as the relationships between the species and the environmental variables that you're actually interested in testing. And so what we have to do is we have to permute the residuals of our model. And so what we do, and there are two ways we can permute the residuals of the full model. So this is where we have, and I've got these things back to front relative to the matrices. I knew I would do this. These are back to front. This really should be X if you follow in the arguments. So this would be X and this would be Y over here, okay? But anyway, we have our response variables, we have our matrix of explanatory variables, and we have our matrix of conditioning variables, and then some residuals. Well, in this model, we can permute the residuals, put it back through our model to generate new data, and then fit our model to that new data, but that when that would generate our null distribution, and that preserves the relationships among all of these things. Or we can do it on the reduced model here, and there's there we just fit a relationship between the response variables and the partitioning or conditioning variables. We get the residuals, we can permute residuals, and then add them on to the fitted values of our conditioning to get new data, and then we fit the model to that, and we keep doing that iteratively, okay? Now the idea here then is that these two models maintain the relationships between the species and the conditioning variables, or between the species and the environmental data in a sense, right? And they keep the relationship between the environmental data and the partitioning variables as well, all right? And there's a nice paper by Kayata Brack and Yari Oksanen and Pierre Legendre a few years ago in Methods in Ecology and Evolution, which goes into a lot of detail, and I just realized I haven't about these methods, and I realized I didn't put it on the slide, and I should have done, so I will add that because it goes into a lot of detail to explain what's going on here. Okay, if we're doing permutation tests, we need to have a test statistic, and we compute that for our observed model, and we compute that for each of our permuted models. So be it whether we've permuted the species data and refitted the model on that permuted data, or if we've permuted the residuals and then refitted our model on that permuted model, right? Permuted data. So we're going to generate this test statistic, and the test statistic that we use is a pseudo f-stat, and it has a form similar to the f-statistic in regression. So we have the chi-squared of the model, so like the variance explained or the inertia of our fitted model, divided by the complexity of that model, and then we divide that by the chi-square of the residuals, so the variance of the residuals, and we scale that by the complexity of those residuals, how many degrees of freedom were left over in the residuals, okay? And so you can see that if this number on the bottom is small relative to the number on top, then we have a model that will explains a lot of the variance relative to, so it has a high amount of variance explained, a small amount of unexplained variance, then F will be large, right? And our aim then is under permutation to look whether F is unusually large relative to our null distribution. And what I'm showing here then, and I'm showing you the code to do it, but let's just ignore this for the moment. I'm looking at the permutation statistics from a permutation test applied to that first model that we fitted, okay? And so in this case, there were no conditioning variables, so we could just shuffle the species data and it doesn't really matter what we do. So we just permute the species data and refit the model. And we do this 999 times. And what you get out of it then would be an F statistic, and these dots at the bottom here are showing all of the F statistics that we got for those 999 models fitted to the permuted data. And then this is a density curve that I'm showing over those sets. So it's essentially just trying to approximate or estimate the distribution. So this is the null distribution that we've generated using our permutation test. And the line here is being drawn at the observed values. This is the observed value of F. And so you can see there's only a small proportion of the permuted F values that are larger than the observed statistic. And so we see here that it's about 0.35 in this instance, right? So for this test, and I didn't set the seed or anything, so I couldn't reproduce this. But here, the p-value is about 0.35. So it meant that about, about 96% of the permuted F values were lower than the observed F statistic, right? And that's how permutation tests work. We hope that our observed value of F is extreme, is in the extreme upper tail, because F we want to be big. Okay? All right. So that's how these things work. And as we've just seen, the way we do this is using the ANOVA function. So it was ANOVA that was actually generating the permutation test. There's no analysis of variance going on here. It's just how ANOVA is usually used for regression models for testing. So Yari took the same thing. And ANOVA is just an interface to a lower level function called permutest.cca, which is what's actually doing the permutation testing. But ANOVA is sort of the user-friendly interface to that. Okay? And what I just showed you with that test was ANOVA was testing by default the whole model. It's doing an omnibus test whether the whole model is significant or not, right? But there are other things that you can do. And so this is, if you do that ANOVA or apply the permutation test to our model, this time using a seed, a random seed, so we can always get the same set of permutations. Then this is the calculation for the F statistic. It's coming from these chi-square values here. So we get this chi-square value divided by 14. That's the model part. And then we have our residuals here. So this is our residual sort of variance, our residual chi-squared. And this is the complexity or the degrees of freedom for the residuals. Right? That works out to give you this F statistic. And our p-value in this case is 0.029 or 0.03. So it's saying about 70% of the permuted values, 97% of the permuted values had an F statistic that were smaller. Only about 3% of the permuted of the F statistics for the permuted model were greater than the observed statistic. So this ANOVA method has a number of arguments that control what is actually permuted and how things work. And so we have this we give it our object, which is our fitted ordination. Permutations is something we'll come on to in a minute when we look at restricted permutations. That controls how we actually do the permutations. And by default, it's doing 999 random permutations. And then the other key thing here, yeah, I said the argument I said before, it was I thought it was method. It is actually model. Okay, so that's you can control which of whether you permute under the reduced, the direct, or the full model. And then by determines exactly what we're going to test. And I'm going to explain what the different bys are now. So we can do a number of tests. One thing we can do is we can test the overall significance of the model, and I've just shown you that. We can test the individual canonical or constrained axes. So to say maybe we only need the first two, or maybe we only need the first one axis to explain, and the remaining constrained axes are not important or not significantly different from the null model. We can test individually the model terms that were added sequentially to the model, a bit like type 2 sum of squares. I think it's type 2 sum of squares, but these sequential tests that you get with regression. Or we can do the marginal effect. And so the marginal effect is where you test the extra information that comes from adding one variable to model, given that all the other terms were in the model when you did it, and you repeat that for each variable in turn. Okay. And so the most useful one of these really is that, I mean, actually this sequential one can be is less useful unless you have really orthogonal terms, because the order that you put them in the model formula can actually determine how these things, or what the p-values are here. Okay. So the default is the top one, and then you choose from these other three using the by argument. So if we do by axis, what's happening is the first axis is tested, and that's how much variance is explained by that first axis compared to the residual variance. And then we partial out the first axis, and then that allows us to test the next axis, because the second axis would become the first. And then we test that axis against the residual. So we're doing an F statistic, the same sort of F statistic, but we're only using one axis at a time. And as we go to the second and third and higher constrained axes, we partial out the lower axes. So if we want to test the third axis, we'd partial out the first and second axes. And so what we're doing here is looking at how much extra information can be explained by these later constrained ordination axes. All right. And when you do that for the mods variable that I had, sorry, the mods thing, I think this was the forward selection thing, then we had three axes because we had three variables. The first two of those constrained ordination axes are significant. And so really when you're doing your ordination plot, maybe you only need to show the first two axes, so a triplot of those first two axes. And this third one, it has to be there because we had three sort of variables, but it's not really explaining much variation relative to the others. And so maybe you don't need to explain that, okay? Or interpret it in too much. If we have by terms, this does this sequential ordering from first to last. And so it adds in it's a bit like forward selection, but it's doing it in terms of the order that you put the variables in the formula. So it tests the effect of adding the first variable in the formula to the model. Given that's there, it now has a model that contains that first variable, and it says, okay, how much extra information can I get by adding the second variable from the model formula in, and so on. And so you do this using by equals terms, unless you've got purely orthogonal terms, and that can be hard to achieve in practice. Then the ordering of the terms will affect the result. So if you just change the ordering of the variables in the formula, you'll get a different result, which isn't very helpful. Okay, so terms in itself is not that helpful in many situations, but it can be useful if you have a proper sort of experimental design and you set the model up properly with the right contrasts and so on. Okay, so here I'm just showing it for the same model. This is our forward selected model by terms. Aluminium was the first thing that was selected, so and you know it is highly significant. Then we have phosphorus, and then we have potassium. And now because this model was forward selected, we would expect these permutation tests to also tell us that these things actually had significant effects in the model. But if I'd done this on CCA1, the the shoot yourself in the foot model I fitted at the beginning, then there at some point these things would stop being significant. But if I just reordered them and put aluminium last, aluminium might come out as having a non-significant effect. Okay, so the ordering matters here. If we do type equals margin, sorry, by equals margin, then what we get is the marginal effect. And this is, I think, is more useful. It's given all of the terms that you have in the model, how much of the conditional on those variables being in the model, how much does the individual term contribute to that model, given that we already have some things in the model. Okay, and this is more like the t-tests that you get in the summary from a linear regression model. And so this is what we're doing here, because those t-tests in the summary from a linear regression model are the effect of these variables given the other variables in the model, so like the partial effect, for example. Okay, and so here we can see that if for the line for aluminium says, if we have a model that contains the other two, phosphorus and potassium, how much extra does aluminium provide? Well, aluminium provides quite a bit. That's the chi-square is 0.31, so that's quite a lot. Given the total chi-square here, and so it's highly significant. Okay, then the line for phosphorus is saying, if I have a model with aluminium and potassium in, how much does phosphorus add? And likewise the same for potassium. If I want to test for potassium here, that's it. We have it relative to a model that contains aluminium and phosphorus. Okay, so each line in here says how much, what is the additional effect given that I have all the other things in the model already. What is the additional effect? So what is if the first line, what is the additional effect of aluminium given that I have phosphorus and potassium in the model? And, you know, again, as we would expect, given the forward selection, these things are all significant. Yeah? And I've only done this here with this forward selected model just because it meant that I don't have stuff going off the slides at the bottom. Okay, I think the fact that you've already done some forward selection will mean that these things are significant. So you'd be a bit careful about sort of garden or forking path kind of ideas and what have you here. Yeah. Okay, so I'm going to have a quick example now looking at the spring meadow vegetation. And so this is an example, and the data themselves are also taken from this nice textbook by Jan Lepsch and Petra Schmilova. It's the second edition, I think. Yeah, the second edition of their multivariate analysis of this book here, multivariate analysis of ecological data using Kanoko. I'll put the reference up. Now, even though it's using Kanoko, which is commercial software, it's really got a load of useful information in for fitting these kinds of models, lots of example data, and so I've been trying to replicate some of the analyses in their book using Vegan. Okay, so to do this, we're going to load Vegan, and this is where we're going to load data from the repo. And so that's why you have to be running it in the base of that zip folder that you just that you downloaded and extracted, because it's going to load data from the data folder. Okay, and they're just CSVs. There's a little bit of cleaning up we have to do. We have to tell it we've got some headers and that the row names are in the first column and stuff like that. Okay, we're going to load this into the species data into an object called sp and the environmental data into an object called env. Okay, and just move myself out of the way. Okay, so CCA is a good starting point here because there's a very long gradient in these plant data. There's lots of species turnover over the particular data set, the set of sites. And if you're interested, you could do DCA using the Decorana function to go and look at the effects of these things. Okay. Now I'm just going to do the very silly thing at the beginning, which is to just throw in all of the environmental variables. So I'm doing CCA, I have my data frame of species, and I'm going to throw in all the environmental variables that are in this environmental variable data frame called env. And then if we do a permutation test on this, I set my seed, that's the main thing, and then I test the significance of this model. So I use the ANOVA function for that. And this tells us that somewhere in amongst all those 15 explanatory variables that there does seem to be something going on, right? So our task now really is to sort of try and decide if there's a smaller subset of those variables we can get away with using those. And so we plot the data. This is the kind of thing that we get. And we get this really long gradient. we get this long gradient, so we have this sort of arch configuration in the sites. That is a real arch, I think, with these data. You wouldn't want to detrend it away. This is a long species gradient. And these are the explanatory effects. Now, I don't think it's helped. This arch is not helped by having all of these predictor variables in here, but, you know, the arch is actually real. Okay, so just be careful with this. I think the arch is real anyway. Okay, so that's our base model where this might start from, and this is just a look-see. But again, ideally what you'd want to do is maybe, rather than just throwing everything in, maybe you've selected some of these variables first based on your hypotheses. I'm going to assume that these variables were collected for a particular reason, and that's why they all went in here. Okay. So now I can do some the sort of forward selection, maybe using the ordistep function to sort of choose a smaller set of variables. All the problems and the caveats that we've talked about stand here. Okay, and when we do that, and I'm setting the seed to make this reproducible because this is using permutation tests, then with 7 of those 15 environmental variables, we can explain about 20% of the variance in the data. And that seems to be perhaps all we need. Okay. And there's a lot of structure in this data. The first constrained eigenvalue is very large relative to some of the others. So the first axis is going to be one that contains most of that variance explained. In fact, it's going to explain about half of it is going to be on that axis, just given by the size of this eigenvalue relative to this number here, the constrained. Okay, and there also is some quite strong patterns in the residuals. So the first and second, well, and even the down to about the fourth or fifth unconstrained axes all explain more variation than the second constrained axis. So even with these 7 variables, there's still quite a lot of structure left over in the residuals. So now we plot this. Doesn't really matter that the thing has been flipped, okay? The fact that this is flipped upside down doesn't really matter. It's just the sign on the eigenvectors is not defined. So you can, this configuration is the same whether it's flipped this way or that way, or this way or that way, okay? There's the same configuration, so we can't tell a difference. And some computers will have the arch going this way, and some will have it going the other way. But these are the effects. And so we see here we've got ammonia and the organic content seem to be high in these sets of samples, whereas the pH and what have you and the conductivity are higher in this direction. Conductivity sort of seems to be more, sort of highly, conductivity and calcium increasing this way. It seems to be most important on this first axis, with the organic carbon and ammonia on the other axis. Iron, the amount of iron in the soil, for example, perhaps reflecting the soil type, it seems to be more important on the second axis, for example, here. Okay. And so if we look at that forward selection, we see that calcium was the first thing to go in, then conductivity, then the amount of organic C in the soil, and so on. And pH was the last thing that got added. And then an alternative that we can do is, because that arch, if we were worried about the arch showing up just because it's an artifact in correspondence analysis, it can be an artifact in correspondence analysis, but also just to show you that we don't have to use CCA, we can use RDA for species data. If we transform our data first here by applying a Hellinger transformation, then what RDA is doing is actually doing is preserving the Hellinger distance between samples, or the chord distance between samples, in terms of their species composition, and that sort of tends to work relatively well with species rich data. And so you can use RDA to get an ordination that's similar to the sort of CCA thing you get. And if you do that, you end up actually with the same model, or the same variables in the model, I think. Okay, maybe some slight differences, but they're going to be sort of showing the same sorts of things. Okay, and if we look at the plot, we get a similar sort of configuration. And the reason that all the species are all crammed up down here is just the kind of scaling that's been used in this plot. All right, we could spread them out a lot more, okay, by choosing a different scaling. Okay, so and if we do the stepwise using the adjusted R-squared, so this is the approach that Guillaume Blanchet and colleagues proposed. If we do that, then we actually get slightly different things, and it says, okay, so calcium and ammonia, and this is using it, I think, from the RDA, I think I've done here. Then we get calcium, ammonia, conductivity, silica, organic carbon, nitrate, and then pH. Okay, and then at that point you got over the global R squared term. All right. So that's just showing you some examples of fitting this, and you can do this in your own time and explore these data in a bit more detail. Okay, I'll have a quick look at the Google Doc, and then we need to get on and look at some restricted permutations. Okay. Okay, somebody's already asked about set.seed. I'll talk about whole plots and split plots later. How do you determine the seed? Oh, you just put any number in there. Yeah, but it's just you need to tell it a number, otherwise you won't get the same. And if you do want to have the same different permutations each time, then you can just don't set the seed. But the thing is then is if you haven't set the seed, you'll never reproduce your results. And so if you're doing this for a paper or something, you need to set the seed, because what that does is it sets the random number generator off in a certain starting point, and then if you ask for 999 random values from that point on, or permutations, then you get the same set of 999 random permutations each time you run it with that seed. So as long as you set the seed and then run the test, if you want to run the test again, you go back and you set the seed, then you run the test, you'll get the same answers. Okay, so let's have a look at restricted permutation tests now. And this is important, but I'm going to go relatively quickly through some of this stuff, because this stuff's here mainly for notes, but I just want to explain what's going on when we do this, and then show you how you set things up. So we've looked at what we're shuffling. So we were shuffling usually we either shuffling the species or we're shuffling the residuals of some model, and we talked about that already. Okay, but now I'm going to talk about how we permute something, and these two things together determine whether this is a valid test or not. All right, and really important is how, because a lot of the time if you don't have any conditioning values, conditioning variables, then it doesn't really matter whether you're permuting the species or the residuals, and in fact vegan would by default just permute the species data. But the how part, what we are actually, how we do that randomization will totally determine whether the test is valid. Okay, so all the tests that I just showed you were using complete randomization, and that assumes a null hypothesis that says that all of our observations are independent. Okay, that there's no sort of relationships between them. But if we have temporal or spatial correlation in our data, we have clustering or repeated measures, we might have a nested sampling design with whole plots and split plots, then that won't be true. We might have blocks of samples that we don't want to exchange observations with. Those things all need to be taken into account when we do our permutation test. If we just randomly permute time series data or clustered data or data that have been collected in a certain experimental design, then those observations aren't independent anymore. There is dependency. And so we have to shuffle the data in a way that maintains that structure and that dependence. Conditional on the model. Now, Kanoko for a very long time has sort of been one of the leaders in this area. It had a large number of restricted permutation tests that Kaya Tabrak coded up. Vegan has recently caught up. We're not quite there. The Petra, Schmiller, and Kayo keep adding extra little things. So we were on parity, and now there's an extra thing I think in Kanoko 5.0 that we haven't got in Vegan or in Permute yet, but we're getting there. Now, traditionally, Vegan only used to be able to do complete randomization or complete randomization within blocks via the strata argument. And the strata argument is still present. It's just that now we have this permute package that I wrote that grew out of some code that we were adding to the Vegan repository to add these restricted permutations. And then we realized actually that other people might want to make use of these restricted permutations. So I ripped the code out of Vegan, put it into the permute package, and then worked on it there. And a few years ago, we got Vegan and Permute working together very nicely, so that, as you've already seen, I've shown you there's this, the permutations argument controls how we do these permutations, and that is using all of the functionality from Permute. So I'm going to go through some of those restricted permutations and show you how they work, and then and show you some examples. Now, permute is loaded. It's a dependency of vegan, so it will actually be loaded and available for you. So you can use the functions within permute without loading it explicitly yourself. Okay, so what permute is doing is very closely following Kanoko, and it's done this because Kaya Brack was chiding, in his very friendly, nice way, complaining when permute didn't do something he thought it should do, and asked me to fix it and to add some new stuff. And so we've made it work like Kanoko because that's how what people were familiar with at the time, what I was familiar with, and so on. So the key thing here is that we can think about our samples as belonging to 3 levels of a hierarchy. We have the sample level, so that says and that's where we define how individual samples themselves get permuted. Then we're going to have a plot level, and these are how samples get grouped at an intermediate level. So plots can contain samples, but the plots might be permuted if we wanted to, okay? And then we have an outermost level, which is the block level, and this tells us how samples get grouped at that most outermost level, okay? Blocks contain groups of plots, and each of those plots can contain groups of samples, okay? So we have these 3 levels of the hierarchy. So the key thing to remember, though, blocks are never going to be permuted. So if we define blocks, only the plots or the samples that only the plots or samples within the blocks will get shuffled. We never swap samples between blocks. So if you define blocks, then they fix everything, and the permutations are only going to go on within those blocks, never between them. So that's a key thing to remember. We can have plots or samples within plots, or we can have both. These can be permuted. So you can permute the plots, you can permute the samples, and keeping the plots fixed, or you can permute the plots and the samples if you want to sort of randomize all of that stuff depending on the hypothesis. And how we actually do the shuffling can be in terms of pure randomization. We can shuffle them according to a time series or a linear transect for equally spaced data in time. Or we can have a spatial grid for regular spacing in space. We can permute, as I've said, we can permute the plots themselves, so we can swap around entire plots of samples, okay? Or we can keep them fixed. And so we have these 5 options, and these things can be applied at the different levels. So we can have multiple plots per block, multiple samples per plot. Plots can be arranged in a spatial grid. The samples within the plots might form a time series, okay? You can get quite complex setups here. But even with all this complexity, it isn't actually enough to cover all of the sorts of things that ecologists do. Okay, so let's look at the different levels. So blocks are some random factor that does not interact with the factors that vary within the blocks. So these are things that perhaps they're a nuisance variable, but you've had to just set them up. You might have plots in different mountains, you might have plots in different fields, and there's no real difference between them. It's just that you've got these 3 different blocks and you want to account for the block level variation, because observations on block 1 are going to be more similar to one another than observations on blocks 2 and 3. All right, so you don't want to shuffle between them because they're not independent, right? So as I said, we never permute samples that between blocks, only within. And this is what you could achieve before using the strata argument in vegan, and it had to be a factor variable, okay? Now the key thing to remember here is that variation between blocks needs to be excluded from the test, but you can't do that with permutation. With permute, what you have to do is add this thing here, plus condition blocks, where blocks is your factor variable, and then that would remove the effect of the block. So it would remove the between block variation first. Okay, and then the permutation test just does the correct permutation by not shuffling the observations between blocks. If we've got time series or linear transects, then rather than randomly permuting them, we ought to try and permute the ordering or of the samples in time or in space along a transect. And the way we do that is via cyclic shifts, and this diagram is showing a cyclic shift. So what we do is we take all of the observations, and I've got 8 observations in my time series or my transect here, And what we're doing is we just bend them into a circle. Okay, so we bend them into a circle at some starting point. Okay, and then what we do is we randomly choose a starting point. And so the starting point here is shown by the arrow and the red thing. And so we randomly shuffle them in time order. So this might be the actual ordering of the data. If we were to randomly shuffle them in time order, maybe we start at time 0.4. So we go 4, 5, 6, 7, 8, and then we start with 1, 2, 3, keeping them all in order. Okay, but we just joined the ends together. That allows, and by joining the ends together, we can just keep shuffling this thing round at random places and start it off. So here we're seeing, this is the observed ordering, and then we're seeing two other random starting points. All right, so that's the cyclic shift, and that works for equally spaced time series and equally spaced transects. So this works okay if there are no trends or cyclical pattern in your data. The autocorrelation structure only gets broken at those endpoints, and if they're stationary. If they're non-stationary, then this is going to cause problems, okay? But of course, you could always detrend your time series to make them non-stationary, so remove the effects of time, year, for example, if these were yearly observations. But don't detrend your time series to make them stationary if you want to test for the significance of a trend, okay? But you can do that. And I'm just showing you, here's another, this is how the numbers actually work out. We joined the sample, there's 10 samples here. We're joining them at between 10 and 1, so that's the join. But we said this random starting point was actually designed, it was 6. So we go 6, 7, 8, 9, 10, and then start at the beginning, 1, 2, 3, 4, 5, okay? And we can just keep generating those random permutations by choosing a random starting point, but keeping the observations in order as we shuffle. Now the same idea of cyclic shifts translates to spatial grids and in 2 dimensions. So we can actually do the cyclic shifting in 2 dimensions. So we can either, and do it at both same time, we can cyclic shift around the toroid this way, or we can cyclic shift around the donut this way, all right? And this gives you random permutations, and actually this random permutation isn't that helpful, but anyway, I maybe change the seed and get a slightly different permutation. But this is the grid, and this would be a random permutation of that grid, okay? And if I just move myself here, when we do that, you need to tell it how many rows and columns your samples are. And you obviously, your observations have to be in grid order, okay? So the next thing to talk about is split plot designs. So we might have, in split plot designs, we have 2 levels. We have whole plots, which contain the split plots. And so the split plots are the samples, right? That's why it's a bit confusing with the terminology, but the split plots are the actual samples, but they are in whole plots, so they're grouped at some level. And you might be applying some treatments both at the samples within, and then some treatments at the split plots, and then some treatments across the whole plots. And with permute, what you can do is you can permute the split plots, or you can permute the whole plots, or you can permute both of them at the same time. The only thing is, if you're going to permute the whole plots, then you have to make sure that the whole plots are the same size, because you're taking this group of observations from over here and you're switching them with another set of observations, okay? So that's the important thing, that the whole plots have to have the same number of observations in them. And so what you might end up doing is, if you don't have balance in your experimental design, you might have to, for some tests, if you're going to shuffle those whole plots around, then you might want to drop some samples. Well, you'll have to drop some samples from some of the plots so that you have balance, okay? So this can test the effect of constraints that vary between the whole plots. So like if there are some variables that only vary at the whole plots. So maybe you've got whole plot A, B, and C, and they have different amounts of fertilizer that were added, and then the individual plots within them might have had other treatments applied to them, but at the whole plot level, it's the amount of fertilizer or nutrients that you were applying, okay? So you can permute the whole plots, and you keep the order of the split plots the same, fixed, because we don't want to randomize and shuffle them, yeah? Or we might test effects of constraints that vary within the whole plots by permuting the split plots within the whole plots, but we keep the whole plots fixed. Okay, so you might fix the whole plots and just shuffle, randomize within the whole plots, and that will test effects of things that vary within the whole plots, not between them. All right. So that's what a split plot design is and how this thing works. The whole plots, the split plots, or both can be any one of these things. They can be time series, linear transects, regular grids, or just random permutations of them as well are allowed. So you can apply those restrictions both at the whole plot level or at the split plot level. Okay. And another thing that you can do with permute is if the split plots are parallel time series, and time is sort of an autocorrelated error component affecting all of the time series, then it would make, essentially what that's saying is that each observation, each observation at the same time, whether they're in whole plot A, B, or C, they're all experiencing the same time. So we should use the same cyclic shift in each of those whole plots to the within ones, right? So we can just apply the same one, and that's using an argument called constant equals true. And if you do that, then this is just more respecting the fact that the observations experience the same time rather than them being essentially at random times. And there's one more thing that we can do. We can use mirrored permutations, and this is just a way of allowing for isotropy, in that just as if you were to go forwards in time, you might be able to go backwards in time. That's the same sort of thing. Maybe if you're on a linear transect, if you walk forwards along your linear transect, that's the same way as if you walk from the other end towards yourself. So we shouldn't just only be permuting one way, we can permute two ways. Now, usually this is more important for linear transects than time, but it applies. And so what we can do is we can shuffle it backwards and forwards. So we choose a random starting point, but we can choose that random starting point by going anti-clockwise or clockwise around the circle. Okay. And so in that sense, the permutation 1, 2, 3, 4 is the same as 4, 3, 2, 1, and that they have the same sort of general idea. And although they are different permutations, they're equally valid permutations under this null hypothesis of isotropy. Right? So, and the same argument can be made for spatial grids. And one of the reasons why you might want to do this is that if you turn on mirroring, if it's appropriate, then it can double or quadruple the number of potential permutations that you have available to you. Because these restricted permutations actually really limit the number of possible permutations that are in the data, and that limits the possible p-value. Okay, so mirroring can really help if you're willing to accept this isotropy thing. Okay, right. Importantly, there's no free lunch here. Permutation tests might sound amazing, that you can just do whatever you want with them and you don't have to worry about the assumptions. As long as you permute the things that according to the null hypothesis this independence. But the main problem with them is that the minimum p-value you can get is 1 over NP, and NP is the number of allowed permutations, including the observed set of data, the observed orderings. Okay? Because under the null hypothesis, the observed orderings are just one of all possible orderings of the data. So, for example, if we didn't have mirroring turned on and you had a time series, 1 sample every year, 20 samples, really long data set, probably took you a long time to collect, the minimum p-value you could get would be 0.05 in that situation. If you had 100 samples, the smallest p-value you could get would be 0.01. So there's a lot of effort to go to end up with a p-value that can barely hit significance at some notional arbitrary standard of significance in science. So if we had 10 data sets with, if we had a data set with 10 time series each of 20 observations, okay, and we want they were all collected at the same time. So we had, let's say we had 10 plots, and we had 20 observations from each plot, and they were collected over 20 years, but the samples were collected from the plots in the same years, same 20 years. If we turn on constant equals true, we had 200 observations in this data set, but under that constraint, there are still only 20 possible permutations of the data. So still the minimal p-value you can get is 0.05. So you have to be very careful that you have enough replication in your data set to apply these so that your test has enough power to detect an effect. Okay. Okay, and if you want to, you can generate all possible permutations, and you can tell it to do that. And often when when there are relatively few permutations possible, so like on this one here where you only had 20 or only 20 possible permutations in these settings, it's better to switch to an exact test where we evaluate all of those possible permutations and do an exact test rather than doing the random sort of test and select them randomly. Okay, and you can force that by doing complete equals true in your call to how. Okay. So what are we going to do then? The way we set up the permutations is with a function called how. And then if you want to do the sampling yourself, if you wanted to use permute in your own code, then shuffle will give you a single permutation from the permutation scheme, and shuffle set will give you back n permutations from that. So there's an argument n. If you want 10 or 20 or 100 or whatever, shuffle set will give you back that set of permutations, so you only need to call it once. And then if you want to get all of the possible permutations for a particular scheme, you can use the allPerms function. All right. Now this was designed for these relatively small data sets in ecology. Allperms is pretty slow if you ask it for a scheme that has many thousands of possible permutations. It'll do them for you, you just have to sit around and wait. Okay. Now all of this is actually built into Vegan now, and so you don't even need to do shuffle and shuffle set and allPerms yourself. All you need to really worry about is how. I'm going to spend a few minutes going through what how works, and then we'll have a quick example, and then I'll move on to doing some PERMANOVA. And I see we're running out of time a little bit, so I'm going to skip the example and go on to PERMANOVA because I know a number of people were interested in that. Okay, so how has three main arguments. It has this argument within, plots, and blocks, and these reflect the different levels. So the within is the samples that are within plots, that's the lowest level. Plots represents that intermediate level, which might be your whole plots, and then blocks represents this overarching blocking factors. Okay, and they have helper functions. So the within function has a within with a capital W, so this just differentiates the argument from the function. Okay. So they're capitalized, and then plots can take this helper function plots, and then blocks can be set up just as a sort of factor variable. So what I'm showing here is I'm just creating a factor that has 3 levels with 10 observations, and so if I wanted to just permute my observations, saying that my plots are grouped, so there's 3 plots, and I want my 10 observations within each plot to be a time series, then this is how I do it. I say that the within permutation type is series, and then I have to tell it that the plots, where the strata for the plots or the definition for which variables goes in which plot is held in the plot function. So plot is a factor, it has 3 levels, 10 observations per level, and that encodes, that tells me which observations belong to which plot. Okay, and now with that permutation structure, we would get time series cyclic shifts within each level of the plots, so within each of our 3 plots. Okay. And so these helper functions make it really easy to change just one or a couple of the aspects of the permutation scheme, leaving everything else alone. And so you see that within has there's things like constant, mirror, the number of columns and rows if it's a spatial grid. But if you don't have any of those, you don't want to change any of those, you just want to tell it that you want to use time series, then you can just fiddle with the type argument, yeah, and leave the other ones at their default. So you only need to change the things that you only need to change the things that you need to change, and that's the reason for using these functions to set up your permutation design. Okay. So how has some additional arguments that control a lot of the heuristics that kick in, that stop you shooting yourself in your foot by trying to ask for 10,000 permutations when you only have 10 possible permutations. So there's an argument complete, and then you can set complete to force it to enumerate the entire set of permutations and do this exact test. And then if you want, you can also say if the minimum possible permutations drop below a certain level, then it would be better to do an exact test in that case. That's a heuristic that we apply. And if you just look here in the arguments, the minimum permutations is set to 5,040, and I can't remember exactly why that is. but it's at some point, if you have less than 5,040 permutations, then vegan and permute will automatically switch to doing complete and exact test, right? And that computes in relatively quick time, doing them this. So that's why there's obviously a bit of a trade-off here between doing the exact test and doing the random permutations. And this is the sort of point where we've decided it sort of makes sense to do a complete enumeration. If you have more than 5,000 permutations, possible permutations, then it makes sense, maybe better to do the randomization, the full randomization. Okay. So let's have a couple of examples and then I'll do PERMANOVA and then we'll finish, okay? So time series example, here we've got 3 plots with 10 observations each, and I've already sort of shown you this a little bit. So we have 3 plots, 10 observations. We set up the control argument with the how, and then you would pass that to the permutations argument in vegan, but here I'm just showing some one randomization of that, okay? And I'm just showing you them. This is plot 1 here. So this is the first observation. Notice it's wrapped round. So actually the starting point was 9. So we go 9, 10, 1, 2, 3, 4, 5, and so on. In this one here, the crossover point is here. That's the join between the 2 seasons. So the random starting point for this plot was at point 14, or sample 14. Yeah. And then in this one, the crossover point is here. Okay. So we're just sort of showing how to do the shuffling. That's what's actually being done under the hood, but you wouldn't need to do that. You could specify that, just pass it the object H in vegan, and it would do all of this for you, right? And then this is the effect of the constant time. So now I'm showing you the constant time, and this is showing you the same join. They had the same starting point. So the things were started, and then we started in the 9th year within each of our plots. So they had the same starting point because the split point is here. Yeah, it's where they joined. So that's the effect of the constant argument. Okay. So I'm going to skip. Actually, no. Well, let's do this because I want to show you how you pass in some of these permutations. It'll take a couple of minutes, and then we'll do the PERMANOVA example. Okay, we've seen how we can drive permute and how to use the how commands to set all this stuff up. Now what we're going to have a look is this ORAS data case study 5 from the Lepschen-Schmiller book. So it's repeated observations of composition from an experiment where there was a factorial design, 3 replicates. The treatments were fertilization, mowing, and then removal of this of millennia. And we're going to test just one of the hypotheses that they had in this particular study. So the first this hypothesis that we're going to test is that there are no directional changes in the species composition in time that are common across all treatments. Okay, so there are no, or the specific treatments. Okay, so there are no common directional trends in any of the species. That's a null thing. The alternative obviously would be that there are trends in some of the treatments. And so we're going to load the data up, and we have to just remove some bits of information in here. So the amount of millennia is stored as one of the species, so we have to pull that out and then remove that millennia variable from the species data. And then year is numeric, and we want it as numeric, but it actually came in as a character vector. So we're just going to convert that to be numeric. Okay, that's all we're doing here. And then if I just remove myself. Okay, so what we're doing here is we're fitting our RDA. We want to look at any time effects. So we want to have this is a global trend, so a trend overall data, all of our observations. And then we want the interaction between year and mowing. So if the site was mowed or not, then that interaction, because mowing is just a 0-1 data, this tells us whether there's a trend just in mowing that's different from the common trend, whether there's a year in the fertilizer treatment, the sites that got the fertilizer or not, versus the global trend. And then here it's the same for the removal. Is there any trend in the removal samples that's different from the global one? So we have a global trend, a trend that's an extra trend that might be in the mowing samples, a different trend that might be in the fertilizer samples, and a different trend that might be in the removals. And then because these are all done at this plot level, we have to remove the effect of the different plots because these are a replicated design. Okay, so there were plots. So we remove the effects of these different plots because that's essentially a nuisance variable. Okay, but we we want to permute them at the plot level, right? So that's what we want to shuffle here. Okay, we don't want to shuffle the data within. We want to keep those observations fixed, right? So we do this test, we don't permute within because the year is varying at the plot level, right? So that's there, okay? And so with the plots, we tell it that the strata are the plot IDs from here, and then we have to tell it that we want to randomize, do 3 randomization, or just randomize the plots. And because these plots have equal numbers of observations, number of samples within each plot, we can do this shuffling of the whole plots. Okay. So when we look at the design here, we have blocks, so there's no blocks here. The plots were defined by the plot ID. What am I saying? There's no mirroring and things like that. We only ask for 199 permutations. I think that's the default. So on. Okay. So we can pass. That's our model that we've fitted. But now when we want to test it, we're going to use this how function, this h object, to tell vegan how to do the permutations. All right. And so we do ANOVA. We give it our model, and now we pass in this h object to our permutations. And I'm going to follow in the book, I'm going to do it under the reduced model rather than the full model. Okay. So when we do that, there are these 4 terms because we had the year effect, we had the year effect, and then the 3 year by treatment interactions. And that says, okay, so this here we would then reject the null hypothesis that there are no directional trends. Okay. Because this value is significant, right? And then if you want to test the individual axes, for example, we can do that by switching up the by term, and we keep the permutations set using this h object. So as long you just have to change the h object, create your own how, that controls how we permute these samples. Okay, I'm going to skip over the hierarchical analysis of crayfish. We can come back to that if people are interested in more later, or just look in the slides. I think it's really sort of self-explanatory what's going on. I can add a few notes to the slides to explain this, but and essentially just going through the same sort of analyses each time. So I wanted to talk now for a few minutes about PERMANOVA. Okay, so what is MANOVA? So MANOVA is the classical multivariate form of ANOVA. We have multivariate response data, we have categorical predictors. We try and decompose the variation in the responses into variations between groups, variations within groups, and we want to test to see whether that second one, the variance between groups, is unusually large relative to some null hypothesis that there is no group effect, right? Depending on, and we have these categorical variables that define the groups. Okay. So doing that test at the end there, testing to see if the variation between groups is unusually large relative to some null hypothesis, requires a lot of assumptions about multivariate normality and all sorts of other things that rarely hold for ecological data. And so PERMANOVA was sort of dreamt up by Marty Anderson, invented by Marty Anderson, as a way to get around this problem. And the perm bit in PERMANOVA means permutational multivariate analysis of variance. So the idea here is that you could do multivariate analysis of variance, which is essentially just like RDA or something like that, but you can use permutation tests to do the analysis and do the testing. You don't have to rely on the theory. Now it avoids most of the issues that we avoid most of the issues with the classical test by doing these permutation tests. And in Marty Anderson's original one, you wouldn't just do RDA, you could, and assume Euclidean distances, you could actually assume any dissimilarity matrix, and her original technique directly decomposed that dissimilarity matrix into variation within and variation between. And then you would do find out how much variation was explained both within and between the groups, and then you'd use a permutation test to test whether the variation between groups was large relative to the null, and you generate the null using the permutation test that we just discussed. So this technique, I mean, PERMANOVA sensu stricto really just applies to this Adonis function. Vegan has 4 different ways to do this kind of analysis. Adonis very much adheres to Marty Anderson's original definition. It's called Adonis because Yari thought it would be funny because it's related to the analysis of similarities like anosim, but this is meant to be analysis of dissimilarities, and so Yari decided to call it Adonis for some reason as a play on those words. So Adonis does exactly what Marty Anderson's original idea was, but the way it was implemented, it only allowed for these sequential tests. And so be very careful with the original Adonis function, because the order that you put your variables into the thing controls whether the things might be significant or not. Adonis 2 is a newer implementation that's actually based on ideas of McArdle and Anderson. It was actually published in the same year as Marty's original Adonis idea, or well, PERMANOVA idea. But Adonis does things slightly differently. And so it's the same kind of model, same idea, it's just implemented differently, right? And you should probably use that because we have much better ways of doing the testing. All those tests, the testing by axes and by margin can all, by margin, not by axes. By margin can be done with Adonis too. We also have distance-based redundancy analysis, which is another technique. This doesn't directly decompose the, actually, distance-based redundancy analysis does directly decompose the matrix, but does it is a slightly different way of doing it and handling of negative eigenvalues and things like that. Cap scale is the classical distance-based redundancy analysis of Pierre Legendre and Marty Anderson. And so this is how the different functions map onto the different papers. As I said, be careful with Adonis. And really one of the main differences between them is how, in terms of what they actually do, is how they treat the negative eigenvalues. And negative eigenvalues just arise because some of the dissimilarity coefficients like Bray-Curtis and other things like that we use in ecology, they aren't metric, and so you get you can't represent them in a Euclidean space because they don't follow the triangle rule, and therefore that you get these variation in the complex plane as well as in the real plane. So the plane of imaginary numbers as well. And so, yeah, probably too much detail, but it's just how they treat those negative eigenvalues. And so this figure shows the general idea of what PERMANOVA is trying to do. And essentially what it's doing, given 2 sets of data, we've got some observations that where we've got multiple responses and multiple samples. They came from these grazed plots, and then there were some where we have multiple species and multiple samples. They came from these ungrazed plots, and we're looking at the data in some multivariate space. So we've summarized this multivariate species data down into 2 dimensions, and now what we want to know is if the mean from one group is different from the mean of another. And we're going to test the null hypothesis that these means are actually the same. And we want to look for evidence that there is separation between these means. That's what PERMANOVA is doing. It's just doing it in high dimensions. And that's why it's ANOVA. Okay? That's all ANOVA is doing, is looking for differences of means and testing whether the observed difference of means of your groups is consistent with there being no difference, the null hypothesis. So that's the general idea. Okay, so if we do this in Adonis, and I'm going to use the June meadow data, so you need to load the June meadow data because it has a factor variable. It has a couple of them actually, I think. Well, no, sorry. The management, the A1 is the depth of the soil horizon. Okay, now Adonis doesn't have to have just factor constraints. It can have linear variables as well. So A1 is a linear variable, management is a factor, and we're looking for the interactions between these two in this model. Okay, and so when we do Adonis, we also have to tell it how we want to do the permutation test. So I want to test the individual terms, and this is actually going to test them sequentially. So it's going to test the effect of management, then the effect of the A1 horizon, and then their interaction. Okay, and when we do that, and because we didn't set anything for the permutations argument, we get 999 random permutations, and the permutations are being done randomly. Okay, so there's no restrictions. So we see that management is significant. The A1, so that's a factor because it's got 3 degrees of freedom here. The A1 horizon is also significant, but the interaction term isn't. Now that could be because of the order in which these things entered the model. So there's another test that we can do, which is to look at the margins. And so if you reverse these things, for example, we get different significances for the A1 and the management. The interaction comes last in this case, but there is a way to test that, and that's to look at the margin. And that's what I'm doing now. So on the previous one, I just flipped around the ordering to show you that the ordering mattered. Okay, now I'm going to test the margin. And when we do that, we only get one row. So these are the marginal terms. And there's only one term here. The interaction term is the only term that isn't marginal to another one, i.e. this term, management colon A1, doesn't occur in any other terms in the model. Management occurs in this higher order term because it's in the interaction, and A1 is present in the interaction. So we can't test their effects. So the vegan actually does the right thing for you and says, okay, the only thing you can test is the interaction term in this model, or the only thing that makes sense to test. It tested it. It's not important. Okay, so we could maybe leave off the interaction. And if we do that now, if we leave off the interaction and just look at the two main effects, then we see that they're both important, and we still see that management is the more important, perhaps the more important effect than the A1 horizon. All right. Now, if you were just doing this in the strict sense of Marty's PERMANOVA original one, then you would really only have these factor variables in. But just in the same way that ANOVA is just an ordinary linear model, it's just that you've got these categorical constraints or predictor variables. In the same way in regression you can mix categorical and linear predictors, we can do the same thing in PERMANOVA. Okay. Now, there's one big problem that we have to be aware of, and I could have done that with db-RDA, with distance-based RDA, I could have done it with cap scale. They all would have worked in exactly the same way. It's just you might have to use the ANOVA function to do the actual tests for those, rather than them being built into the Adonis functions themselves. And I've used Adonis 2 here because Adonis 2 is the one that allows for these different types of testing by margins or for the whole model. Right, so there's one problem that we have to be very careful of, and that's the dispersion problem. So even in Marty's original paper in 2001, she was noting that the method can confound location and dispersion effects. And so what's happening here is that if one or more groups are more variable, i.e. they're more spread out around their centroid than than the other groups, so it could be one group or several of them could be more spread out than the others. If there's a difference in that dispersion around the centroids, then that can result in a false detection of a difference of means, even if the means are all the same or essentially the same. So even if all of the groups were lying right on top of one another, if one of the groups was more spread out, that could get misinterpreted as a difference of means, when really it's just a differences of variance. And this problem affects the t-test, it affects ANOVA the same way, because to more or lesser extents, t-tests and ANOVAs are a little bit more robust to this, but it can still affect them, okay? And it's the same with PERMANOVA. And so there's several papers here. So Dave Wharton drew some attention to this a few years ago, and colleagues with their paper in Methods in Ecology and Evolution. And then Marty Anderson and her colleague, and then there's a more recent paper on this problem. We've actually looked at what's going on when you do have differences of dispersion. Now, there's not a lot you can actually do about this in vegan other than to test whether you have differences in dispersion. And so what we mean by dispersion in multivariate space is the spread. How much are your points spread out around the group centroids? Okay? So we can see here there might be some tendency for the grazed plots to be more spread out in multivariate space, more dispersed than the ungrazed ones. Yeah. In 2006, as part of some work that Marty Anderson was doing on biodiversity sort of metrics as well, Marty came up with this test for the dispersion, this PERMDISP. So it's a multivariate test of multivariate dispersions, or differences of multivariate dispersions. And the general idea here is that you can calculate how far each observation is from its group median. So let's say these are the group medians here, and the little grey arrows that are joining, the grey lines that are joining the points here, they represent the distances between each observation and the median. Okay, we can take those distances, we take the absolute values, because some could be going in this direction and some could be going in this direction, so we don't care whether they're positive or negative in that sense. Okay, we just want the absolute values. And then what we would do is just do an analysis of variance, an ordinary analysis of variance on those absolute distances within groups. So you can see if you have a group that's got larger absolute distances, i.e. that has larger grey segments on average, if the average of those length of those grey segments is higher in one group than the other, then that can tell you whether you have a dispersion problem, a difference in dispersion. Now what Marty Anderson was doing it for initially was to sort of say, well, these sites here are more diverse because they're more spread out in terms of their species composition, say, than these sites. But this idea that she had, actually she had 2 papers in 2006, this idea that she had, there's this, the one I'm showing here is from her biometrics paper, which actually explains the test itself, all right? And then we test the null hypothesis that there are equal absolute distances to medians among groups. And you can do this in beta disper, and it's called beta disper because part of why I originally wrote beta disper was because I was using it for multivariate diversity analysis, and so I implemented it from that viewpoint. But now it's also obviously useful for testing for dispersion effects in Adonis or distance-based redundancy analysis and so on. Okay, so we can do that, and what I'm doing here is I'm just going to take that lichen data, and then I'm going to create a Bray-Curtis dissimilarity matrix of the lichen data, and then I'm going to artificially put on whether the sites were grazed or ungrazed. And I forget now whether this is actually true or not, whether they were grazed or not, but I'm just going to say that some of them were grazed and some of them were ungrazed. Okay? And then with beta disper, what you do is you pass in your dissimilarity matrix that we created here, the Bray-Curtis, and you also pass in your factor variable, right? So we just take a single factor, and then we can test to see whether they're the null hypothesis that these two groups have the same variance. And then when you do that, this is the model that you get. So this is actually kind of like a model, but there's no test done here. This is just showing the average distance to median in the two categories, grazed and ungrazed. And it's telling us that there is, it's higher in the grazed than in the ungrazed. Okay? So this is the grazed. These distances are higher on average. That's the median distance to centroid in the grazed, so it's higher. And this is the median distance to centroid in the ungrazed. Actually, it's the median distance to the median, actually. Okay? We're using the median as a robust measure of the centroid. Okay? And then this is it. This is this plot. And so that thing that I was showing you to illustrate was actually what PermDist is doing. It's looking at these multivariate distances, and we use the permutest function on our model to do a permutation test and see whether this observed difference is consistent with, actually it's the sort of thing you would get if there was no difference between them, or whether this represents evidence of a real difference between the groups in terms of their dispersions. So what we're essentially doing here is now randomly allocating the sites to the two groups. We rerun the PermDist test to get the average distances, and we compute the distances between them. And actually what we do is we gather up the F statistic in the ANOVA, or essentially, we gather up the F statistic from the ANOVA that we're doing. So we calculate that, and then we generate a random null distribution for F, and we look to see how often our observed value of F is exceeded by the values from the null distribution. And it works out to about 4.5% of the time in this instance that the null distribution exceeded our observed data. So in that case, we conclude at the 95% level that there is a significant differences in the deviances, sorry, the dispersions of these samples. Okay. And you can do this pairwise as well. So we can look at if you had more than just, so this is an omnibus test over the groups. And then this is a pairwise comparison between the things. These things don't necessarily have to give the right answer because this is a test on F and this is a test on something else which I can't exactly remember, but they don't always give the same numbers. The p-value below the line, this is based on the classic p-value from the ANOVA, and this here is the p-value that's based on the permutations. So by telling it to do pairwise comparisons, you can actually work out which groups are sort of significantly different from one another and see where the dispersion problem really is. But you could probably also get that just by looking at the plot as well. Okay, now we don't really have a solution to this dispersion problem. Marty Anderson and colleagues in this 2017 paper have proposed a solution that's actually related to this Behrens-Fisher problem in economics, I think. Now, from what I understand with people talking about this, this is only available, this new thing that Marty's come up with, invented, is in Primer. It's not yet in Vegan, and there is an outstanding bug report for us to try and add it. I haven't even looked at the paper yet to see what's involved and whether it's going to be easy to do or not. So we can't do anything in Vegan to solve it. Primer has this particular thing here, but Marty's 2012 paper also talks about what you're actually testing if you do have differences in dispersion when you do PERMANOVA, and so that's a good paper to look at. Okay, so I'm going to skip over my other stuff. I've got a couple of things at the end I just want to mention. So in terms of the links, I've actually got some lots of examples and things for using vegan on my blog. Go and look at this link here to see those different posts. You can have a look at the ones that relate to vegan up there. I will actually put up some more links to some other resources in the slides this afternoon. And I just want to thank NSERC for funding me in my lab. And just want to shout out to Yari, Oksana and Peter, especially Yari, who done us a great service by putting all of these years of effort into developing vegan. Peter and I help Yari maintain vegan. Peter's done a lot of work on the community sort of simulation and the null model side of vegan with Yari. I've mainly stuck to the ordination side of things. But you know, I just want to shout out to those guys because they help us maintain this software. And also thanks to all the other authors of vegan who've contributed stuff over the years. I really appreciate the efforts that people have put in. Okay, so I'm going to go back to the Google Doc now, and I'm just going to see if there are any questions here that I can go through. Yeah, so we don't have anything really to address this issue of reduced power in unbalanced designs. I mean, ideally you should solve that by getting your design balanced in the first place. So we can't do that and there is no modification, I don't think, for this modified f-test in there, so we have to be a bit careful. We haven't looked at bootstrapping, and there is no bootstrapping sort of available in vegan for any of these things. But that could be an option. We haven't really explored that in vegan. The idea, I think, would be you could do this yourself, but you'd have to extract. So you could use vegan to fit things in your bootstrap. So you could use the vegan code, but you would want to turn off doing any permutations. So set permutations to be 0, and then you could wrap that in your own bootstrap code, and you could do that. We can partition the permutations within groups, and so you would use blocks for that. So the permutations are only done within strata or blocks, the block level, sorry. And you can do that using permute. You just have to set up the permutation design using the how function. So you just need to set the blocks variable to be a factor, and then the permutations would only happen within those blocks. So question here, I thought I might get this. How do methods such as CCA and PERMANOVA compare with JSDMs, these joint species distribution modeling approaches to doing ordination? And so if you haven't heard of these things, we have things like Borel or HMSC that are actually fitting proper statistical models that do the kinds of ordinations that CCA is trying to do, or RDA. So how do they compare? Well, I mean, Borel and HMSC don't suffer the same sorts of problems that CCA, but more that the distance-based techniques can get wrong. CCA isn't perfect, but it does a better job than some of the distance-based stuff. Borel and HMSC will do a lot better potentially, but they also have their own problems and things. And so I think we're moving towards things like Borel and HMSC and joint species distribution modeling approaches. But they're much more complicated to fit. They can take a lot longer to fit. We haven't quite, I don't think, I mean, we just spent, I was just at the Statistical Ecology, the International Statistical Ecology conference a couple of weeks ago virtually, and there was lots of discussions about how we interpret these things, how we fit them, how is there any, what's the advice now that we've had sort of 10 years of these things maybe, that, or not quite 10 years, maybe 6-7 years of these things. What advice can we provide? I mean, so we're still working on understanding these, and I think we have a much better understanding of CCA, especially in RDA. And we know how PERMANOVA works, and we know where it fails, essentially. So yeah, ideally, I think maybe we would be using Borel and HMSC. I think they will run into problems with sample size, just in terms of being able to compute things physically on your computer practically, whereas CCA and so on would not have any problem dealing with the sort of with large data sets. I think that, you know, more likely to encounter problems using Borel or HMSC. Although as computers get faster, as we get more memory, these techniques become more accessible to larger and larger data sets. I think we're going there. I think also when you might choose one over the other is actually something that people are still discussing, and actually the conference, there might be some output from that conference, a working group that might be writing a paper, I think, on some advice as to what situations, what do they do, where are they most useful, where might you have a problem with CCA or PERMANOVA that you might want to switch to using Borel or HMSC. Okay. Is it possible to run PERMANOVA in vegan with a hierarchical nested design? Yes, you can do the whole plot, split plot. Well, in fact, you can do any of the the restricted permutation tests. The Adonis and Adonis 2 and what have you, and the ANOVA function, if it applies to RDA and cap scale and things like that, they all take a permutations argument, and so you just set up the permutation design that you want for the nested approach, and then you pass that to the permutations argument, and then vegan just knows, it understands what that means, and it will just permute the data in a nested way, respecting that hierarchical way. We can only do 2 levels of nested design, so we can only have samples within plots within blocks, right? That's what we can have. We'll just have there's 3 levels. We have the lowest level where you have your individual samples, we have the plot level, and then we have the block level. And so if you can fit your data into that kind of design, then we can do that. And often very nested designs can actually be accommodated by just not having, like if you've got multiple levels of a hierarchy, each level represented by a different factor, there might be, depending on what test you want to do, there might be a way of representing it as one factor versus another. So you have these 2 levels, okay? Even if the actual design might have 3 or more in. So you can do that. Some caveats, because I don't know exactly what level of nested design you're talking about. But I think if you've got just 2 levels in the nest, that should be okay if you use blocks and plots. Okay. Yeah, is there a function within vegan that implements Anderson's CAP paper? I'm not 100% sure what you mean by that. I think CAP scale is the thing that I thought implemented CAP. And actually it turns out to be very similar to the distance-based redundancy analysis of McArdle and Anderson, which is a bit different again, I think, from the distance-based redundancy analysis of Pierre Legendre and Marty Anderson. I think we've gone through a lot of iterations. Well, not me, but Yari has done a lot of this work, just trying to harmonize cap scale, DBRDA, PERMANOVA, and how they all, and the Adonis functions and how they work. And actually now cap scale, it turns out, is just doing what DBRDA does. You just turn off how it deals with the negative eigenvalues. And so yeah, Yari has talked about deprecating cap scale, but we haven't done anything, and we probably won't. I mean, we might not do it. I don't think there's a reason that we should deprecate it, because people have written cap scale code, and that still works. There's nothing wrong with it. It's just how the main difference really is just how it approaches those negative eigenvalues and whether it ignores them or whether it tries to correct for them or whether it actually uses the imaginary distances. And PERMDISP and beta disper, for example, they don't necessarily correct for the negative distances. They can use those negative distances and we can bring them into the calculation. So I think if you have a high proportion in negative eigenvalues, that's basically telling you the amount of distortion that's going on, trying to take this non-metric dissimilarity matrix and squish it into a metric space. Okay, so if you're using one of the techniques that does try and squish it into this metric space, then the more negative eigenvalues you have, the higher the distortion, and therefore the worse that mapping in the Euclidean space is going to be. And so I can't give you a a sort of rule as to how many of them there, but there are, like the distance-based redundant dBRDA, for example, in vegan, can do the same kind of analysis that PERMANOVA is doing, but we can correct for the negative eigenvalues or use them, right? And take account of the fact that there are distances in the negative in the imaginary plane as well as in the imaginary numbers. So the complex plane rather than in the real plane as well. And so we can use that information. So maybe using dBRDA is better than using cap scale from vegans' point of view. Okay. And there are ways. So I talked about ways of adjusting the to get rid of the negative eigenvalues in the webinar on Tuesday. And so you can use the Lingo's method to correct for the negative eigenvalues as well. And I think you can do that in dBRDA. Okay. Does it matter whether the data are unimodal or linear for PERMANOVA? No, it doesn't matter at all, really. So what's happening here? We have these two different ordination techniques, the linear group, the PCA and the RDA, and then we have the unimodal group, the CA and the CCA. And that they were categorized that way because their underlying response models being linear or this unimodal species packing model. What PERMANOVA or an Adonis or distance-based redundancy analysis are doing is they're saying if you're more interested in that situation, you're more interested in what the dissimilarity coefficient represents. So if it's a Euclidean distance, that will be getting distances between your observations in terms of the abundant taxa, whereas the chi-square thing in CCA and CA, that treats all species the same, and so the rare species get weighted, for example. And so there we're looking at relative composition. So whatever features of the dissimilarity metric that you use, whatever feature that dissimilarity metric has, then PERMANOVA will decompose that information. So if you have a dissimilarity coefficient that works on relative composition, then that's what you're getting. You're getting a test for differences in the means species composition where you're looking at relative composition effects. If you use something that works on the abundances like Euclidean or Manhattan or something like that, chord, then you're looking at differences between the sort of absolute distances. Differences, absolute abundances rather than the things. Please explain the use of perm beta disper. So I didn't really use perm beta disper. I used the permutest function. So the permutest function is what does the permutation test on beta disper, and it's in terms of the permutation test. It's if I just go back up. So we have I'll do it on, well, this one's a bit clearer here because it's showing the whole screen. So when you do the beta disper, so we've got it here. This is beta disper. This is taking the dissimilarity matrix and representing it in principal coordinate space. So it's trying to do this Euclidean embedding, but we get negative eigenvalues and we get positive eigenvalues in this case because there is it's Bray-Curtis, you see here, right? So we have 15 positive eigenvalues and 8 negative. So there's some non-Euclidean component in here. But we can use, and what I said, you know, we use these negative distances. I can look at the distances in these negative eigenvalues, and I can look at the distances to centroids in the positive eigenvalues and then add them together and in some way, and that's what we get here. This is for the distances to centroid in the grazed group and the ungrazed group. But there's no test has gone on here. This is just calculating the distance to centroids for each group and doing the embedding, all right, in Euclidean space. Then when we do the permutation test, what we're doing is we can evaluate, we do that ANOVA. So we're testing whether the difference in the, this graph here is showing the absolute distances to the centroids, to the medians actually, of those groups. So it's showing you the distances from these data points to their group centroid, the absolute values of these grey lines, right? Okay. And we want to know, this is the observed difference, okay? And we can calculate that using an ANOVA, which is essentially just a t-test when there are two groups. We get this difference between them, that's the observed difference. We can calculate whether, using a t-test, whether that's a significant difference in their median, in their mean distances, right? So we can calculate that, and then we can test that. But because we might be violating a lot of the assumptions of the ANOVA, we test that using a permutation test. And so this is the F statistic from that ANOVA. That's our observed F statistic from the ANOVA. And what I do now is that we just randomly assign observations to the two groups. So I might be taking some of the observations over here and I shove them into this group, and I take some of the observations over here and I put them into this group. So I randomly assign them, keeping the same number of observations in the two groups. I don't suddenly make the grazed group smaller in terms of the number of observations than it is in the data set. So all we do is just randomly allocate the observations to the two groups. We redo the ANOVA for the difference in the mean distances, and we get a new F statistic. And that's one observation under the null distribution. So we do that many, many times. We get lots and lots and lots of values from the null distribution of this statistic. And then we say, okay, how many of those values of F from the null distribution exceeded our observed value, which is this one. And about 4.5% of these 999 exceeded this. And because that's less than some notional threshold, then we would say that this was significant at the 95% level. Okay, and that's what that Permi test is doing. Okay, let's go back to the questions. I don't think PermDist assumes that the data are not standardized. But I think if you try, it depends what you mean. If you try and standardize by the species variances themselves, could you still get observations that might have more variance in one group or not? I think you might be able to. So, yeah. I think if you're gonna, I don't think you should start rounding your species counts or modifying your data in any way. I think you're better off leaving them as they are. You might apply a transformation to the species data before you do the dissimilarity matrix calculation, but I don't think you'd want to start rounding them and things. I think that might just make things worse. Well, so it's not really a question of whether it's not appropriate to use PERMANOVA for data where there is a heterogeneity of variance. The problem is that if you do have a difference in variance in your groups, then you can't conclude from the PERMANOVA alone that there is a difference between your groups in terms of their means, in terms of their species composition, right? And so if you can't say that there is a difference in means because you also see the difference in variance, then it kind of defeats the point of doing the test, right? You could estimate it, but you just can't say for certain whether that if you do find a difference, if you do see significant differences in the species composition, you can't say that that's due to differences in species composition. It could be differences in variance. Marty's 2012 paper talks a little bit more about this, and hopefully, maybe we'll get a chance to get that correction or modified version that's in the 2017 paper implemented. If it's easy enough to do, in which case then we could probably correct the PERMANOVA for that. Okay. I think to a lesser extent, things like CCA and RDA, I think if I remember from Dave Warden and colleagues' paper, they were less affected by the non-constant variance, this heterogeneity of variance issue. So that might be another option to try as well. But I really can't remember 100% the detail of David's paper now. Not 100% sure of what you mean in this question about the trajectories. So if you want to clarify that, I can maybe come back to that after. So what's the difference between RDA and distance-based RDA? So in RDA, we we just take the raw species matrix and do redundancy analysis on it. And I'm not going to explain exactly what redundancy analysis is now, but it's what we do. In distance-based RDA, what you do is you turn, well, there are several different ways. One of them is that you could transform your data and then do, so like transform using a Hellinger transformation or a chi-square, and then do distance-based redundancy analysis, and then do a redundancy analysis on that transformed data. And that ends up getting a redundancy analysis which shows like the Hellinger distances or shows the chi-square distances, for example. Another interpretation of what distance-based redundancy analysis is to take the raw matrix, convert it into principal coordinates, and then sort of decompose it that way. And so there is all these different advantages and disadvantages of these. Well, not advantages and disadvantages. There are actually different things about what distance-based redundancy analysis is, and it's got slight variations. I would have to just go and check exactly what the dbRDA function is doing in vegan, just to confirm. But what are the advantages? Well, in RDA, if you just do RDA on the raw data, then you get Euclidean distances. And Euclidean distances are dominated by the big things. So if you have very abundant species, then those abundant species will dominate the ordination. And so you'll just see differences in the most dominant species, and the rare things will disappear. If you, with the distance-based redundancy analysis, either by transformation or by just giving it a direct dissimilarity matrix to work with, then you can represent whatever you want in the same way, like with PERMANOVA, you choose any dissimilarity coefficient that suits your need. Are you interested in relative composition? Do you have mixed data and you might be using Gowers' mixed coefficient? Then the distance-based RDA and PERMANOVA can both work on those dissimilarities directly, whereas RDA is restricted to basically working on Euclidean distances, or things that you can represent as a transformation that then you apply Euclidean distances to. The strata argument I don't think would help with the heterogeneity of variance things, because we don't necessarily keep the observations within the plots together. Okay, actually no, we do. I still don't think it would help, because you're still going to have the same spread, right? And really, in fact, so it won't help, because the strata is going to be the argument that is the group, and the group is the thing you're trying to test, right? So because it's basically, do you have more spread in one group relative to another, or several groups relative to other ones? And in PERMANOVA, you're actually trying to test that grouping variable, so you wouldn't know. Strata wouldn't help at all. Okay, someone said there might be a few more questions on PERMANOVA. Oh, things are moving around. I saw something. Can restricted permutations be used on the paleoecological time series, i.e. where we don't have equal spacing? Well, it would be better. Yeah, you can, although it's not going to be perfect. You have to be a bit careful about it, so you can do it. It'll keep the ordering correct, right? But obviously, if you're doing the permutation test and you're shuffling one thing relative to another, it's going to be matching up things that are not the same time difference apart, right? because the observations, when you cyclic shift one relative to the other, you're not just moving the same number of lags. Okay, so no, it's not correct 100%. You can use it, and I know people have used it, but you just have to say that, I think you would treat that then as the test is probably a bit too liberal, because it's not really doing the right thing, and therefore maybe you don't use 0.05 as your significance level, maybe use 0.01 as the significance level or something smaller, okay, so that you don't maybe make the false positive. I've seen a paper looking at sort of phase scrambling or something like that. There was a paper in Methods in Ecology a few years ago on sort of different kinds of permutation tests. I think the idea there, they were using it for space, but of course time, we can just treat time as a one-dimensional spatial thing. So I do have a thing on my long to-do list that was to go and look at that paper and see if I could bring that idea into vegan or permute, if possible. Okay. Yeah, okay, so I can't see any more real sort of questions here that were not handled elsewhere. Let me just see if there are any more. Okay, so there's a recommendation for there's a recommendation. Well, not a recommendation. What are my recommendations for if there's collinear variables in your environmental data set? Well, I mean, if they're really strongly collinear, then it's basically telling you that you don't, if you have 5 very collinear variables, you don't really have 5 unique bits of information. Maybe 3 of them can explain the other 2 perfectly, or at least very closely. And so if you just keep including the same things in the model, you might over account, you might double account for some of those effects. But you can also cause problems in terms of, you might inflate the coefficients for one set of variables because you're trying to offset the coefficient that's being estimated for some other variable. And so what often happens, you can have large coefficients for highly correlated variables, and it's just because they're trying to offset the effects of one another because these things are correlated. And so you don't want to, that can cause the same problems. It can make the coefficients bigger, and because the coefficients are bigger, it can look like they're more important, when really they're not. They're just highly correlated, and only 1 or 2 of them are actually important for the in terms of explaining variation in the species. So, or the responses. So how do you deal with that? I mean, would use the VIF function to identify them, and then, as I mentioned at the time, you can just use correlations and the correlation matrix and look at the correlations between things. And then you just decide, if you've got a bunch of variables that are highly correlated, you can decide to remove some of them and throw some of them out. So maybe there are some variables in your data set that you're not that interested. They just came off the mass spec or the ion chromatograph or whatever, that just came back from the lab, and maybe you're not interested in them, but they are correlated with other variables in the data set. So you could get rid of those things. Obviously, if you've got a high VIF and that's a variable that you want to include in the model, you wouldn't throw that out. You'd keep that in, but you'd need to find out which variables were correlated with that variable so that you could remove those things, so that you can keep the thing in the model that you wanted to test. Right? How do we determine how to aggregate data when 2 data sets, community and environment, are collected through several months and years? So the way to think about that is the rows in your data matrix would represent a sample, each sample. So if you go out for several months over several years, every time you went out into the field to a particular location, to a sampling location, that becomes a sample. That is a sample, all right? So you just have them as other rows. So you might go back to the same sample each month and for 5 years. And so that sample would be, that sampling location would be represented by 60 rows in your data set, because you went every month for 5 years, okay? And then in your environmental or your constraints data set, I would have a column then that had the year. And so you'd have in what year each of your observations was taken. And then I would also have a column for the month. And then just store them like that, okay? And then you've got your species data or your response data that you can ordinate. And then if you needed to, you could remove the time effects, for example, by partialing them out by using your environmental data set that has this year variable and the month data, okay? So you just arrange it in the same way. It's just you need to think about it as being a sampling location or a sampling occasion. Every time you went out to sample at a specific location, it has a different row for every time you did that, okay? So what about if you're trying to see if your species composition is moving towards that of a target site, which has a different plot size to your plot? I'm still not 100% sure what you're meaning. I think one thing I would do there, or if I understand properly, it would be to ordinate all of the data, right? And then you can then use some of the tools that I talked about in the introductory vegan webinar, where you might group your observations that this sort of target site or observations from the target site. And then what I would do is I would join up in time how the observations from on the diagram, I'd join them up in time and see how the site is moving, if your samples are moving towards that target site. That would be one way to sort of have a look at that. I'm not sure I get what you were trying to correct for in the analysis. Okay, so yeah. Can you use NFIT for categorical environmental data? Yep, you can. I talked a little bit about NFIT in the intro vegan thing, but you can do that. So in the same way that we can include categorical variables in our constrained ordination models, we can do the same thing with NFIT and include them. And so yeah, you could easily do that. What NFIT does then is it just finds group averages on the axis scores for the different groups, and then it puts in a centroid for them. Okay. So it's not that we can't. So just a confirmation here about what I was saying about if you've got if you're trying to do PERMANOVA and you run the PermDisp test and you find you've got differences in heterogeneities between your groups that you're trying to test with PERMANOVA, there's a confirmation here. Does that mean we can't provide a significant test? We can do the significance test for the PERMANOVA, but the question is what does it mean, right? It might mean nothing. So basically you might find that there is a significant difference in the means in PERMANOVA. But if you know that those groups are significantly differences in the variances, then the significant result in PERMANOVA could be due to the fact that your groups really do have differences in species composition, but it could also be due to the fact that they have different variances and they might have the same species composition or similar. So it's not different enough. And so that's the problem. You're in this situation where you don't know. You've got a result that tells you something, but you don't know whether it tells you that they differ in their species composition or whether they differ in the variance around the centroids. Okay? So I think things are getting a bit moody around here, but it says, can we still use your aggregation solution if there are different sites? You mean if you got months and years and you calculated over that one, then yep, you can do. You just yeah, I said if you went to the same site each month and for 5 years, yeah, then you would just have you went to multiple sites in months and years. So each site would have a different row for every month and every year that you visited. Okay. So someone asked, not really relevant, but is there a reason vegan is called vegan? Yeah, and someone's added, yeah, it's vegetation analysis is where it comes from. Yari isn't a vegan. In fact, he likes his meat. He took me to some very nice restaurants when we taught a course in Joensuu. And I think we taught another one somewhere else as well in Finland. Maybe maybe we were in Uppsala together, and he took me to a very nice restaurant there. Yari likes his food. He likes nice meals, and he certainly wasn't a vegan on those occasions. I think also this is perhaps the Finnish sense of humour coming out. Yari, if you go and look at his website or on his GitHub somewhere, he also has a function, a package called Gravy, and Gravy puts the meat into vegan, he was saying. So it's just the Finnish sense of humour, I think, but it also does map onto something for vegetation analysis. Okay, because I mean that's what Yari does. He is a vegetation ecologist. Yeah. Okay. Okay, I can't really see any more questions. If people wanted to put something in the, add a question, if you're still here, add a question, or if I didn't quite answer it, or maybe if you want to explain a little bit more about what you were meaning, I'll try and come back to them. But we've been going now for about 3 hours, so I think I should stop and go and get a drink of water and have a bit of a rest. But just want to say thank you very much to everybody who's attended the webinar. I hope you found it useful and interesting. Yeah, I hope you now know how to use some of these functions in vegan a bit better. Okay. Well, it's been great to host this and answer your questions and interact with some of you. I wish we'd been able to do it in person at the conferences and things, but hopefully this has made up a little bit for that. Okay. I will, as soon as the recording is processed and I've trimmed that first 10 minutes of just to make sure when, at the beginning when the stream was up, just to make sure that everything was working fine, there was just a slideshow playing. Once I've just trimmed that down a little bit, it takes maybe 24 hours for YouTube to process the live stream. And when that happens, I will make sure that everyone is notified through the email that that the recording is ready, you can watch it back. And yeah, but it will take about 24 hours. Okay, so thank you very much, everybody. Enjoy the rest of your week and stay safe out there. Wear a mask. Bye-bye.